Hi everyone, welcome to the online session of the 6th International Astrobiology Conference. For those who have been with us since the beginning, you might recognize me. I'm Zeynep Eşköse, the head of Agenastro, and I'm currently pursuing my PhD in astrobiology at Istanbul Technical University. This journey began back in 2021 during the challenging days of the pandemic. What started as a small dream among a group of passionate students has now become a cherished tradition. From our very first event, which became one of the largest astrobiology conferences worldwide, to this sixth edition, our mission has remained unchanged: to build a bridge between the stars and our world. Over the years, we have had the honor of hosting some of the most brilliant uh, minds in the field, and today is no exception, actually. As a student-led community that started with zero budget but infinite passion, seeing you all here today is deeply moving. Today, we continue our quest for knowledge with four incredible international speakers. And thank you for being a part of our story and for proving that science knows no borders. Without further ado, let's begin this journey into the unknown.
Dünyanın dört bir yanından bizleri takip eden bilimseverler, her sene geleneksel olarak düzenlediğimiz 6. Uluslararası Astrobiyoloji Konferansı'nın online oturumuna hepiniz hoş geldiniz. Bu yolculuk 2021 yılında pandeminin o zorlu günlerinde başladı. Bir grup tutkulu öğrencinin kurduğu küçük bir hayal, bugün artık gelenekselleşen bir büyük buluşmaya dönüştü. Dünyanın en büyük astrobiyoloji konferanslarından birini gerçekleştirdiğimiz o ilk günden bugün ulaştığımız 6. konferansımıza kadar tek bir amacımız vardı: yıldızlar ve dünyamızın arasında bir köprü kurabilmek. Bugüne kadar alanında birbirinden değerli isimleri konuk etme onuruna eriştik. Bugün de bu geleneği sürdürüyoruz. Hiçbir maddi gücü olmayan ama sınırsız bir tutkuya sahip olan bu öğrenci topluluğunun imkansızlıklar içinde böylesine büyük bir organizasyonu sürdürebildiğini görmek benim için gerçekten çok duygulandırıcı. Bugün 4 kıymetli yabancı konuk hocamızla birlikte bilginin peşindeki yolculuğu devam ediyoruz. Hikayemizin bir parçası olduğunuz ve bilimin sınırını tanımadığını bizlere bir kez daha kanıtladığınız için hepinize sonsuz teşekkürlerimizi iletiriz. Sözü daha fazla uzatmadan konferansımıza başlatmak üzere sözü ekip arkadaşım Berra Nur'a bırakıyorum.
Hi everyone, welcome. Welcome to the Agenastro 6th International Astrobiology Conference. My name is Berra Nur, a molecular biology and genetics student at Uluslararası University and a member of the Agenastro team. It's now my great honor to introduce our speaker, known for her valuable contributions to the field of astrobiology. Dr. Velislava Ilieva is an astrobiologist at the Open University, and her research focuses on the terrestrial extreme environments as analogs for early Martian conditions. Now, she presents her talk titled The Lagoons of La Mancha as Analogs for Early Mars.
Hello,
Welcome,
everyone.
Dr. Ilieva. How are you today?
I'm well, thank you very much. I'm very happy to be here, and I'm very grateful that you invited me.
Thank you. Thank you for being with us. If there is nothing you would like to add, you may begin your presentation.
No, thank you so much for the introduction. If you bear with me now, I'm going to share my slides. Sorry.
Let me help. Yeah, we can see it.
Yeah, is it in full screen?
It's not full screen, but it's okay, I think.
Oh, is it not full screen?
Let me check on YouTube because here it's a bit more different. Yeah, it's okay. Yeah.
Okay, great. Yeah, no, thank you very much. So today I'm going to tell you about my PhD project. I recently completed my PhD at the Open University in Milton Keynes, and I was part of Astrobiology OU. So we are the biggest astrobiology group in the UK and maybe in Europe. We need to double check those numbers. But it's been an absolute privilege to be part of the group and to be mentored by lecturers and professors there. And as I said, I'm very happy that I can share my journey with you today through explaining what my project was about and what things I found out in my PhD journey. So to begin, I'm going to tell you a bit about Mars. Mars through time and why we're interested in Mars.
I'm so sorry to bother and intervene, but could you please click the hide button there because it's a bit blocking the. Yeah. Thank you so much. I'm so sorry.
No, oh, good. Sorry. So Mars is very interesting from astrobiology perspective because even though present day Mars looks a bit like a desert, as you can see here on the image on the left hand side of my screen, so there's no liquid water on the surface. It's very deserty like. There is high radiation impacting the surface. There's no atmosphere, and in general it's not hospitable to life. We have gathered evidence over 50 years of Mars exploration, both in situ with rovers and remote exploration from orbiters, as well as satellites. We have landers, and we have seen that Mars actually was very different in the past, and it has a lot more dynamic history than we thought of. So, for example, here at the top of my screen, I've shown you kind of key features of Mars through the ages. So first, if we start with the Noachian period, we can see that we have a lot of aqueous activity. So this is the period that we connect most with liquid water. We have evidence for valley network formation, crater lakes. We see as well precipitation. So actually a friend from the Open University, he is doing a lot of research on aqueous history of Mars, and he identified and recently published a paper about how much rain, actually liquid rain, so we think like water, like here on Earth, has been in the Martian past, which is very different from what we see now. However, something happened with Mars. It lost all of its surface water and now looks like a desert. And we don't know exactly why it happened. Some theories include that the internal dynamo of Mars ceased. So here on Earth, we have a magnetic field that it's protecting our atmosphere. It's making the conditions on the surface much more hospitable to life. And that used to be the case, we think, for Mars as well in the past. But then that internal dynamo in the Martian core stopped. And we think this is what caused the Martian atmosphere to be blown away by radiation and also the loss of surface water. So that now we see desert, but in the past we think there might have been even oceans. As you can see here in the photo on the right hand side, there are features on the terrain of Mars that are consistent with the presence of oceans. However, we can't go back 3-4 billion years ago to find out. What we can do is we can study the geology of nowadays Mars, we can do modeling and try and recreate what we think Mars looked like. So along those lines, I'm going to tell you how we do that in terms of looking for past life on Mars. We have three main methods in astrobiology. We have simulation studies, or you can think of them as Mars in a bottle. This is where we go into the lab and we simulate certain features of Mars. It can be used to simulate present-day Mars. So for example, here on the first image on the left, I've shown the Mars chamber that we have at the Open University. And this chamber can simulate the very low atmospheric pressure on the surface of Mars, as well as the radiation that the surface is experiencing. We also have a light gas gun that simulates impacts on the Martian surface. So I think we can reach velocities about 7 meters per second, which is quite high. And we can study what impacts could have done on potential biosignatures on Mars. The second way is to do modeling studies, as I mentioned before. You can think of modeling as Mars in a computer. Because we see present-day Mars, we can use different types of modeling to recreate what it looked like in the past. For example, here this image shows different minerals, and based on the deposition of the minerals, we can infer what temperature they were deposed under. We can infer what liquids alter those minerals, and then we can kind of say, oh, for example, you needed acidic liquid to make a mineral called jarosite, and we detect jarosite now on the present day surface of Mars. So that means that that rock had interacted with low pH fluid at some point in the past, right? So this is how we go back in time. We can do that with atmosphere modeling as well, and we can do that with crustal formation, and a lot of different physical characteristics of Mars. And finally, we can try and hunt for Mars here on Earth, or this is going to analog environments here on Earth. And this is what I'm going to focus for the rest of the talk, because I also did my PhD on analog environments on Mars. So most of the analog environments are considered extreme environments, and these are environments that are normally have less life than typically. So for example, if you go out in your back garden, soil is not really an extreme environment. However, if you go to Antarctica, in that soil, that will be an extreme environment because you have the low temperature, you don't have as much nutrient inputs. So this is the kind of places that we think represent Mars nowadays and also in the past. And we have different types of analog environments as well. So we can have geological analog environments, and one feature of Mars is that it's very volcanic, rich in volcanic rock. So we can go to environments here on Earth that are mainly covered by basalts, for example, or like lakes that sit on top of volcanoes, and we can study those as analogs. Other types of analogs are also chemical. So this is relevant to places on Mars that we have detected a lot of salt phases, and we think that concentration of salts could have occurred. And we can study this on Earth by going to salt flats or brines, for example. So here I've shown Western Sahara on the right-hand side, and this is one of the sites that we actually have visited with the astrobiology group. And finally, we also have morphological analog environments, and these are that they have features like appearances that look like Mars. An example for these are the yardangs in Kaidam Basin in China. So yardangs are these features that are formed by wind erosion, and we see them on Mars as well, because wind is one of the main ways nowadays that the Martian rocks kind of get eroded away by wind because we don't have water at the surface, for example. So these are different types of analog environments. For my research and for this talk, I'm going to focus on salty environments or salt-rich environments. And this is because, as I said, we have evidence that there are a lot of places on Mars that are rich in salts. This image here, it's from a paper from 2019 for the from the Mars Curiosity team. And essentially they show that Gale Crater once hosted a lake. So Gale Crater is where the Curiosity rover has been exploring since 2012, 2013. And once it hosted a lake, which is absolutely amazing for the prospects of life. However, as Mars lost its liquid water, as I said previously, we think that the water evaporated and that led to concentration of salt phases. So here you can see calcium sulfate rich salts, magnesium sulfate, and that led to possibly the formation of different ponds with different salinities. So here you would have an undersaturated lake with less salts, but you would also have these super saturated kind of brines with a lot of salt. So these environments actually could have been the last habitable environments on Mars. And my colleagues Rita and Michael are actually going to talk a lot more about this in their upcoming talks later today. But yeah, so salty environments are very interesting. They're also interesting for us here on Earth because they host an incredible diversity of life that can cope with such an extreme conditions, because it's not only the salinity, but also quite often these environments are very dry. They're exposed to high UV radiation, and they don't have a lot of nutrients. So all of this is very similar to what we think Mars looked like in the past or certain environments. So for my PhD, we went to the La Mancha region in Spain. So this region is located in central. Oh, sorry, I don't know why it keeps going further. This region is located in the central part of Spain. It's about hour and a half south of Madrid, and it's characterized by being very dry, having really high evaporation rates and really low precipitation rates. So not a lot of rain, but a lot of water evaporating into the atmosphere. And also interestingly, this region features in the very famous novel Don Quixote de la Mancha from Miguel de Cervantes. And I personally grew up with that novel, and I really enjoyed the fact that while I was sampling my sites, I could see the windmills in the background. So it was a really great experience. So this is a close-up of my 4 sites. So they're called Yaguas, Penahueca, Quero, and Lungar. So all of them, when we went, they were completely dry and desiccated, but they had some slightly different features. So if we look at Yaguas, we see that there is some salt deposits on the top, but they are very small scale and not as extensive. At Penahueca, it was a bit different. So we saw a lot of salt precipitated on the top of the lagoon, but we also saw these kind of polygonal features where salt was concentrated at the edges of these polygons and not in the middle. However, at Quero, then all of the surface of the lagoon was covered by salt. It was quite an extensive salt deposit there. And we still saw these polygonal features, but they were different compared to Penahueca. So their edges were raised, and some of the raised edges didn't have as much salt, but a bit more sediment. Whereas in the center of the polygons, there was a thicker salt crust. And then finally at Lungar, we saw sporadic salt deposits kind of across the lake surface. So these are my sites, and I went there with my supervisor, Dr. Michael Macey, who will give a talk later today, and Dr. Felipe Gomez from the Center for Astrobiology in Madrid. So he was our host, and he's actually also on the Mars Perseverance team, and it was absolutely brilliant to have him there and show us around the sites. The sampling was very fun. We obtained a lot of sediment samples, biofilms. Here in A and B, I've shown 2 different biofilm types. In A, we see a subsurface biofilm, which was about a centimeter or 2 under the surface. And then at B, we see superficial biofilm, which is obviously they're a bit different. And the sample types that we obtained were salt crust from the top. We obtained sediments with a corer. So corer essentially is like a long metal tube that you stick in the sediment, and then you take it out and you get really nice sealed cores. And in the lab, we then go and open up those plastic packages, and we can see here at the middle image how insanely cool was the core. You had a very clear difference with depth. And also, yeah, for quite a few sites, we saw this kind of variation with depth. So we separated those cores into gradients. And then also we collected biofilm. So with these samples, I wanted to do a very detailed geochemical and microbiological characterization to understand essentially what microbes are living there and how they're interacting with their environment and how they are surviving in these very extreme conditions. So first, I'm going to talk a bit about the geochemical analysis. So there's no need to go into a lot of detail. Here on the left, I've shown some elemental plots of the sediments. And the key thing to see is at the Penahueca center and border cores, the salt concentration is much higher, especially sodium and chloride, whereas with the other three lagoons we have a lot more sulfate. And also I just wanted to reiterate that we see differences between these like border and center samples, hence I've presented them separately here on the plots. But yeah, the key take home message from here is that Penahueca, both cores are saltier and they have more sodium and chloride. So when we did the mineralogy analysis, that also was very similar to our chemical analysis in terms of what we found. And you can see that predominantly at Yaguas, Quero, and Lungar, we have this mineral phase in the purple color, which is called gypsum or calcium sulfide, whereas in Penahueca, with depth, so A, B, C, D refers to the depth profiles. So A is top of the core and D is bottom of the core, or E, depending on how many sections we have. We see with Penahueca, those minerals vary quite a lot. So if you remember on the previous slide where we saw salt crystals in the core, so you can see like a tiny image here, but might not be as clear. And sorry, I'm just gonna actually get a laser pointer. So here you can kind of see the little salt crystals. They are all comprised of bloedite, which is a sodium magnesium sulfate mineral. And at the bottom, where we had the orange-red color, we see a lot of clays and carbonates. And the other sides, again, they're dominated mainly by gypsum. They were not as heterogeneous with depth. So next to the microbiology analysis, first I wanted to just introduce the two types of sequencing that I did for my PhD to understand my microbial communities. First, on the left-hand side, we have the 16S rRNA gene amplicon sequencing. It's a bit of a mouthful, but essentially what we do is when we extract DNA from sediments or salts or biofilms, we look for one specific gene, and this is the 16S rRNA gene. This gene is conserved across the tree of life, and especially for bacteria and archaea, it is a great way to identify and taxonomically characterize them, and also identify their relative abundance. For example, here we see that in this community that I've shown, we have 20% bacterium 1, so this can be any kind of bacillus or the Sulfosporosinus or Escherichia or anything like this. And then we have 20% of bacterium 2 and 3 as well, and we have 40% of bacterium 4. So this is the kind of information that we get from the amplicon sequencing. So it's kind of a targeted sequencing approach. And the other approach is called shotgun metagenomic sequencing. And as it sounds, you essentially sequence everything in your samples. So you sequence all of the DNA, you do not target any specific genes or microbes. And this is why we have all of these different genes coming from very different bacteria. We can still do some taxonomic analysis with this type of sequencing, but what is it's actually most important for is to do metabolic reconstruction of pathways. So for example, we can detect how microbes are, what kind of genes they have to, for example, fix carbon, or do they eat carbon heterotrophically, or do they use chemolithotrophic pathways to extract energy from inorganic chemicals. All kinds of questions that we can ask with reconstruction of metabolic pathways. And we can also reconstruct genomes of those microbes without the need for cultivation. So this is very important because it allows us to kind of bypass laborious time in the lab where we culture one microbe by one microbe. We can reconstruct those genomes just from the sequencing data and we can study them. You can also do metagenomics guided cultivation, so we can find microbes in the sequencing data and then we can try and cultivate those in the lab. So it's very kind of valuable way of characterizing your microbial communities. And also we can do like gene abundance kind of investigations where we can see what genes are important for the microbes in these environments to survive. So this is the kind of data. First, I'm going to talk about the amplicon sequencing or the targeted approach. So this is kind of a typical taxonomic profile at the phylum level. One thing to note immediately is that there are a lot of things. So there is a lot of different bacteria and archaea in my samples, which is surprising because it's a very extreme environment, but life still persists. And those lines at the top are a type of a beta diversity analysis where I just compared all of the different communities between the sites and between depths. And the key thing from this slide, the takeaway message, is that we have two major groups. The first group over here, it's all Penahueca communities from salt and sediment, and the salt communities and biofilms from the other sites. Whereas on that second group, you have only sediment communities from Longar, Yaguas, and Caro. So we start to see now in the microbiology data as well as in the geochemical data that Penahueca is slightly different compared to the other three sites. It has higher salinity. The communities are different in terms of composition, but we don't know why. So the next steps would be to use statistics and modeling to identify why is Penahueca different. The first type of analysis I did is called unconstrained ordination. So this is where, very similar to that plot in the previous slide, you compare your communities by the number of species and the identity of those species. And those plots, A and B, essentially mean as when communities are closer together, they are alike. In the Bray-Curtis plot, this is just a distance method that takes into account the number of different species and their identity, whereas the weighted unifrac also takes their phylogenetic distance in account. But again, key thing from these two plots are the Penahueca communities are grouping together away from the sediments presented here with the little stars. And after I've created those plots, I added post hoc these environmental variables. So these are the concentration of different elements that I presented earlier, but I related them to the actual microbial communities. And we see that the Penahueca communities are associated with the concentration of sodium, as well as chloride to some extent, and sulfate here. And here it's even more pronounced. You have sodium and chloride. So we see that this must be very important for those communities and for how they look like. The next type of analysis is called constraint analysis and is a type of modeling. It's a distance-based redundancy analysis. So they look very similar to the other plot, but the difference is how you construct those models. So in here, I've taken into the model both the microbial data and the geochemical data. And then I've created the plot together. In the previous, I added the environmental variables after I created the plot, but here they are added together. And we can see the same thing. The Penahueca communities are associated with sodium and chloride, and they group close together compared to the rest. So now we have found all of this. However, we're still not clear why that is the case. We see that they're different. The Penahueca communities are different from the rest, and we see that they are linked with sodium and chloride. So is it something about the salt or is it something else? In order to kind of tease apart further that story, what I did is performed ecological no modeling. And this sounds a bit fancy, but essentially is using how related are two species, and how similar their niches are to kind of make ecological assumptions. And the underlying assumption for this analysis is that species occupying similar niches will be more closely related than expected by chance. And the way you can think about this is that, for example, if you take two microbes from an Antarctic pond and an Antarctic soil, they probably will be very similar to each other and will be more related. So because they are experiencing the same stressors. Whereas if you compare a species from an Antarctic soil with one from a desert soil, like somewhere in the Sahara Desert, they would be very different because they're exposed to very different environments. And this is essentially what we are trying to kind of tease apart with the ecological no modeling. Is there something different about Penahueca in that environment that is causing differences in the microbes? In order to kind of do these analyses, you have to go into the analysis with a known definition of your niches and your phylogenetic distances. So in the niche, I've taken the geochemical environment, so that is the pH of my sites, and the concentrations of sodium, magnesium, calcium, potassium, phosphorus, silica, chloride, and sulfate. And this is because these were termed statistically significant variables that affected the microbial communities. And what I've done is I calculated an abundance weighted mean for each species for each environmental variable. Essentially, this is finding at, for example, bacteria 1 at pH 7, is that the best pH for that bacteria? Or you can have bacteria 1 in pH 8, for example. You can have more of them at pH 8. So it's finding those optimum niches for each species. And species here means a sequence of the 16S rRNA gene. And then the second thing you have to do is you need to find the phylogenetic distance, or how similar are two sequences. And yes, the underlying assumption again is that species that are similar in sequence, they would occupy niches that have similar physical-chemical characteristics. And this was confirmed. So I did analysis called mantle choreogram, but there's no need to essentially talk about this now. But that analysis confirmed that that is the case for my sites. And then I can proceed with the ecological no modeling analysis, because this here was just testing the assumption and preparing for the analysis. So now the actual analysis has two parts. In part 1, we look at the phylogenetic relatedness again, and in part 2, we look at the similarity in taxonomic composition, and this is between communities. It's a form of beta diversity analysis, and we're going to go through these parts now. So essentially, it's all of this is done by a programming package in R. So I used R for my statistical analysis, and the package does all of that for you. But here, just the steps are broken out for a bit of easiness and to explain them, of course. So how we do is we construct a phylogenetic tree from two communities. And then we see how close these communities are by calculating a beta mean taxon distance value. And then we create this normal distribution plot, and we see essentially the standard deviation between the expected and the observed distances. So the expected distances are calculated by essentially like randomizing that phylogenetic tree at the start to get beta MNTD null. And we have also previously calculated beta MNTD observed, that is for my data. And then with the no distribution, we create this, and then we see how different the observed are from the null. And the key thing is that if two communities are closer than expected by chance, so they're more related. They are affected by homogeneous selection, or this is where the environment affects them so much they're constantly under selective pressure that they're very similar. If the value is above 2, which in this case means that they are more distant than expected by chance. This means they are affected by variable selection. If we go back to the example with the Antarctic soil versus the desert soil, if you compare a community from Antarctic soil with the desert soil, you would expect that variable selection would be affecting those two because they are very different environments. But if you compare two communities from Antarctica, for example, you would expect that homogeneous selection would be affecting them. So this is the kind of inference that we're trying to do. And the next step is to essentially, for all those communities that kind of fall within the node distribution, we do a second type of analysis where we look at their taxonomic composition. And because these communities were not affected by either variable selection or homogeneous selection, they would be affected by stochastic or random processes. So these are processes that just naturally occur in the environment, such as life-death events, so microbes dying, microbes, appearing, or for example, microbes traveling up and down the sediment column, or microbes not being able to travel because of some physical obstacle. So this is very much like ecological theory. And after all of our complicated calculations, the output of this analysis is very simple. So we have those bar graphs. And if we look at A, here I've compared communities within each site to each other. So I've compared all the communities from Yeguas to each other, and I've done that for all of the other sites. And what we see is that for Yeguas, Quero, and Longar, random processes dominate these communities. So random processes are in blue. So homogenizing dispersal, this is an ecological process where communities or microbes can travel up and down, for example, the sediment column, or they can travel between salt crusts. And then we also see dispersal limitation where maybe some of those microbes cannot travel all the way. So for example, if you imagine a sediment, a microbe that sits at the bottom of the sediment at 30 centimeters might not be able to travel all the way to the top of the sediment or to the salt phase. So this is also shown in the analysis. And we have ecological drift. Ecological drift is those random life-death events. However, you can clearly see that for Penahueca, it's quite different. The communities from Penahueca are much more closely related than expected by chance or by kind of node distribution, and they are affected mainly by homogeneous selection. This is where the environment is exercising like excessive filtering on those microbes. And we also have homogenizing dispersal, is where those microbes can travel between different communities. In order to confirm my finding, I also compared the communities across the 3 sites, so Yeguas, Quero, and Longar, and I saw that mainly when you do those comparisons, it's dispersal limitation is the main process that affects them, and that means that the microbes cannot travel from Quero to Yeguas or to Longar, which makes sense because these lakes are about like 10-20 kilometers apart. But when you add Penahueca to the mix and you add those communities, you see that actually now variable selection is affecting them, which means that the Penahueca communities are very different to those at Yeguas, Quero, and Longar. So this analysis essentially showed me that Penahueca communities experience higher salt, and that is making them more closely related. That is exerting active selective pressure. So there must be something happening at the site that is not happening at the other sites, and it's affecting the communities. So now we're going to talk a bit about the shotgun metagenomic sequencing. So as I said, this type of sequencing is where you analyze all of your DNA in your samples. And this is very crucial for reconstructing metabolic pathways, for identifying how your microbes are surviving, or at least the potential for them to survive in these extreme environments. And one thing you can actually do is you can tell which survival strategy those microbes are using by looking at what genes they have. And this is especially relevant for halophiles. So these are salt-loving microbes that we know live in these highly saline sites. But there are two different main strategies for dealing with the high salt. One is called the salt out strategy, which is common in maybe like moderate halophiles or kind of salt tolerant microbes. So this is where they synthesize compatible solutes inside their cell that keeps the salt away from the cell. So kind of they're equalizing the concentration. The second strategy is called salt in, and this is typical for more extreme halophiles where they essentially have halophile specific pathways, and they produce proteins that can work only under high salinities, and they actually pump ions into the cells because they need the salt for their proteins, for their machineries to operate. So by reconstructing metabolic pathways, we can tell what strategies are present. And here on the right, I've shown essentially all of my samples and a bunch of pathways that we're not going to go into detail. But the key thing is that there are evidence for both salt in and salt out strategies. That means that both extreme halophiles and moderate halophiles are residing in my communities. And some of the salt out strategies for compatible solutes synthesis are ectoin biosynthesis, glutamate biosynthesis, trehalose biosynthesis. So all of these things that they essentially synthesize to keep inside the cell, to keep the salt away from the cell. And salt in strategies are certain pathways that are specific for halophilic archaea. So this can be, for example, the semiphosphorylative Entner-Dudurov pathway or pentose phosphide pathway in archaea, and a few others. So yeah, I learned a lot by doing the metagenomic sequencing. And the next thing, now that we kind of know what strategies these microbes have to survive in the salt, we can try and find out where are their energy sources. So how they are essentially making energy, what they are eating. And to do that, I analyzed the biogeochemical cycling genes. So biogeochemical cycling refers to the cycling of key elements such as sulfur, nitrogen, hydrogen, and one carbon compounds. We're going to go into a bit more detail into each. So for sulfur biogeochemical cycling, this is highly relevant for Mars because we have found a lot of sulfur compounds in different oxidation states that can be used for energy. So for example, here, sulfate reduction, these are all genes that are involved in the sulfate reduction pathway. In this pathway, microbes take electrons from, for example, hydrogen or organic chemicals, and they reduce sulfate. So sulfate is an end product of their energy, it's the end is the electron acceptor of their energy metabolism instead of oxygen. So it's a type of anaerobic metabolism. And this is also why it's relevant to Mars, because Mars does not have as much oxygen as we have here on Earth. So microbes that perform sulfate reduction have been identified as like targets for potential life on Mars. And we see that I have those essentially genes in my samples. We have them here at Yaguas at the bottom, and we have them at Cairo. We don't have as many at Penahueca, or like barely any. And at Lungar we have some, but mainly we have them in like down the core, so at depth. We also have sulfur oxidation. So sulfur oxidation is the opposite of sulfate reduction, and this is where microbes use sulfur chemicals as electron donors. So instead of using organic chemicals, they can be quite often they are aerobic, but they can be anaerobic as well. And they here at the UGOS, we actually see that from A to D, so this refers to about 0 to 20 centimeters depth, microbes are using sulfur compounds to obtain energy from them, and they're switching to sulfate reduction at the bottom, which is quite interesting. Another thing to note is here this SQR gene that it's present almost in all samples. This is a gene where sulfite, so it's a form of reduced sulfur, is used to obtain energy, and sulfite has been detected on Mars. So we see that kind of those microbes are they use strategies that we think could be relevant for Mars and very interesting for astrobiology and Mars. The next type of biogeochemical cycling is the biogeochemical cycling of nitrogen. So nitrogen, again, you have similar pathways where microbes can take energy from nitrogen compounds or use them as electron acceptors. So they respire nitrogen instead of oxygen. And it's a bit less relevant for my sites as well as for Mars. We do not see a lot of like nitrates on Mars, but we see here from Yagoas, for example, we have the most. Interestingly, these types of metabolisms are inhibited with salt. So actually the Yagoas samples had the least salt compared to the other sites. So here you can see Penahueca, you have barely any nitrogen related genes. Next, these are all genes that are code for hydrogenases, or these are enzymes that essentially can extract electrons from hydrogen, and we see that they're quite widespread. So hydrogen seems a very important source of energy in these communities, and we know that globally here on Earth, hydrogen is becoming much more recognized source of energy for microbes than it was previously thought. And finally, for one carbon compound, this is also very relevant to Mars because obviously Mars has carbon dioxide rich atmosphere. And we see that, my kind of samples are, my microbes are able to utilize carbon monoxide and carbon dioxide as well as formate. So formate hasn't actually been detected on Mars, but it was good to see that it was an electron donor. So this is kind of about the energy generation of my microbes. And I just want to reiterate with the next two slides about the power of shotgun metagenomics, and that's the reconstruction of actual genomes. And we call those genomes, we call them metagenome assembled genomes, or MAGs. And in these two phylogenetic trees, those with the colored boxes are the genomes that I reconstructed from my sample. So you can see there are quite a lot. On the left-hand side, we have bacteria. On the right-hand side, we have archaea. And what we can also do, because I can go into each individual genome and see what these microbes are using and how they're living, I can find the same genes that I just talked about and pinpoint them to specific microbes. So this is actually absolutely incredible because, I can see that I have here 4 different genomes relating to this species of sulfur oxidizing bacteria without actually culturing those in the lab. And I see that I have sulfur reducing bacteria as well. And what I actually still wish to do is to use some of that data and go into the lab and try and cultivate those different microbes. I have managed to cultivate a species of the sulfur vermiculus, so I have this in culture as well as reconstructing its genome from the data. So just shotgun metagenomics is really, really powerful. However, going back to my thesis in my project, all of my data shows that Penahueca is different and it could potentially be drying up. So this is supported by the higher salt concentration, by the differences in communities, and all of the statistical and ecological modeling that we saw. And interestingly, there is literature, I found literature out there to support that hypothesis. So Laguna de Tires, which is about 4 kilometers away from Laguna Penahueca, has completely desiccated in 2015. And there are studies that I found that apologies that show that there is disruption of the La Mancha region with climate change. So looking at groundwater table elevations, groundwater is one of the main sources of water in these lagoons. Apologies. We see on those plots, like compared to the 1970s or 1990s, in the 20s, in the 20th century, the water table is below the base of the lagoon, which means that it can no longer be recharged by groundwater. And also we see negative trends in precipitation, so mainly evaporation and less precipitation. So that would mean that I'm so sorry, I'm a bit sick and it's coming up now. Laguna Penahueca could be an excellent analog for the transition to a more arid and saline environment, which is highly relevant to Mars, has important implications for the region, ecological, environmental, and cultural, because it's an important region. People go there for bird watching. It has a great diversity of flora and fauna. So its perturbation, it's quite sad and devastating. But based on these findings, we actually, Michael, Rita, and I, and Felipe managed to go back in 2025 to resample the lagoons. So we're now currently analyzing that data and we're going to compare it with the data from my thesis. Thank you very much for listening to me, and I'm happy to take any questions.
Thank you for your presentation, Dr. Ilieva. We can now move on to the question session. I'm waiting for questions. Okay. Our first question is, are there any ancient microorganisms living in the lagoons that could resemble potential Martian life forms?
I wouldn't say that there would be a lot of ancient microbes, because there's still some there's groundwater that's been kind of influencing those lagoons and like moving between the lagoons, and also there's quite a lot of human activity around them, so they've been impacted by agriculture as well as sewage, unfortunately, but they have been protected in recent years under UNESCO, so now they are protected sites. But I would be very skeptical that I don't think there will be any Asian microorganisms there.
Okay, the other question. How does the location of salts affect environmental habitability?
So did you say the location of salts?
Yeah, I said location of salts affect environmental habitability.
So salt, actually, microbes are really good at dealing with salt. Rita later will talk about microbes that actually live in salts, like salt inclusions. So I think the location of salts maybe wouldn't have as much effect. It's mainly about the composition of the salts, because some salts can be chaotropic, so they can disrupt living systems and cells and proteins, where some salts can be cosmotropic, so they wouldn't affect microbes as much. For example, magnesium chloride is normally cited as a chaotropic salt, whereas magnesium sulfate is much more pleasant for life.
Okay, the other question is, have you found anything surprising in the lagoons that change your Mars ideas?
It actually made me more keen on looking into sulfur cycling microbes as a possible model of life for Mars. We have actually at Astrobiology OU, we have welcomed a lot of different guests from NASA, as well from the European Space Agency and different astrobiology institutes. And quite a few of them also like the idea of those sulfate reducing and sulfur oxidizing microbes as models for life. So I think this is the key thing for me. And also recently, I don't know if you've heard about the leopard spots at Jezero crater. The two minerals that they identified in those spots, one of them is a byproduct of the activity of sulfate reducing bacteria. So I think they are very, very interesting for life on Mars.
Okay. It's very good presentation, dear Velislava. I have a question. If Mars had similar Legos in the past, do you think life could have started there?
I don't know about the origin of life. That is a completely different field. It's way beyond me. But I think origin of life would require maybe different conditions, maybe more dynamic environments. So something like hydrothermal vents, where you have a lot of water movement, you have heat, you have different compounds and things like that. But I think salts are really good at preserving life, so they could have been like the last habitable environment rather than the first.
Okay. Other question is, can we design genetically durable bacteria in case of climate errors? I don't know.
Sorry, do you mind repeating the question?
Yeah, of course. Can we design genetically durable bacteria in case of climate?
I think it depends what you want to do. So there is a lot of studies now interested in seeing how climate change is impacting natural environments and how that is affecting the microbes, because microbes are very key for the biogeochemical cycling that I mentioned in the past. So they produce, for example, different carbon gases such as methane and carbon dioxide, but they can also kind of serve as a sink, so they can eat those gases or kind of fix those gases. So I think it can be done, depends on what your question is and what your goal is.
Okay, other question is, may I ask about Laguna, the Penahuca microbiological and geochemical system? Is it relevant to Mars' atmosphere of fauna?
I think it wouldn't be relevant to the Martian atmosphere because it's very different to the atmosphere that we have here on Earth. However, thinking about those microbes that were in the sediment, they are protected from oxygen and they live in very anaerobic, like kind of anoxic environments. So that might be a bit more relevant to the Martian atmosphere. But also on Mars, we are very interested into life in the subsurface. So we are hoping the ExoMars rover Rosalind Franklin, that is scheduled to someday, hopefully in the 2030s, reach the surface in Mars. And that rover has a 2-meter drill, so we'd be able to actually dig down into the surface, and it would be great to see what we find out.
Okay, other question is, if Mars had similar lagoons in the past, do you think life could have started there?
Oh, I think we answered that question.
Yeah, I think so. Okay. New question is, how does seasonal change in the lagoons affect their value as Mars analogs?
Yeah, that's a brilliant question. And this is actually something that we're looking into now. So, because those lagoons are ephemeral, I actually didn't mention that, but during the winter months, they get a lot of precipitation and usually they have water. However, it's not a lot of water. I think usually it's under a meter for sure. I think it's about 20 to 30 centimeters, but climate change has affected this. So this is where Penahueca can be really interesting Mars analog, because if it's aridifying, we can follow the geochemistry, we can follow the microbes through the years. So I really want to go multiple times more to keep resampling Penahueca and at different seasons to see exactly how they would change.
Okay, and our last question is, could surface-based microbes survive beneath the Martian surface today, where water and protection from radiation might still exist?
That's an absolute brilliant question as well. However, we do have percolorates on the surface of Mars today, and we do have quite a lot of studies showing that percolorates are, bad for life. I know there are some microbes that tolerate them, but I think that might be another stressor potentially for the Martian subsurface today. I think as well, we don't know what kind of, as I said, we haven't really been deep into the subsurface yet, so we don't know what minerals there are or what sources of nutrients and energy there could be. So I think quite a lot of people still kind of hope for that idea, and I think it might be possible, but yeah, we just have to wait and see.
Okay, thank you. We finished our questions. Once again, thank you for this excellent presentation and valuable insights, Dr. Ilieva.
Thank you so much for having me. Enjoy the rest of the talks. See you.
Thank you. You're welcome. See you. We will now take a short break, after which our conference will continue from where it left off.
Hello everyone again. Welcome to our second session. My name is Rita Carvalho. I am a second year biology student at Ege University and a member of MBG Türkiye Agenastro. I will now introduce our next speaker. It is my pleasure to introduce Rita Carvalho Serra. Rita is a first year PhD student in the Astrobiology OU group at the Open University in the United Kingdom. Her research focuses on microbial experimental evolution, particularly in identifying genetic adaptations that may be relevant to the habitability of Mars. She will be presenting her talk titled Fluid Inclusions as the Last Habitable Environments on Mars. Please join me in welcoming Rita Carvalho Serra.
Hi, thank you so much.
Welcome. How are you?
I'm good, and you?
I'm good, thank you. You can start whenever you feel ready.
Okay, so I'm going to try to share my screen.
Okay.
Okay, entire screen. Okay. Can you see my slides?
Yep, we can see.
All right, everything looks all right? You can hear me okay?
Yes.
Okay, thank you so much.
Thank you.
Thank you for the invitation, especially because I'm an early career researcher. Just a little context about me. I am a biologist through and through. So I did my bachelor in cell and molecular biology, and I did a master's in biology of extreme environments. And now I'm doing a PhD in astrobiology at the Open University in the Astrobiology OU group. And my PhD project is called To the Lifeboats: Fluid Inclusions as the Last Habitable Environments on Mars. So, as Veli already mentioned before, Mars has looked a little bit like this illustration for the last 3 billion years, in the sense that it's dry, it's arid, and it doesn't really have a thick atmosphere, which means that the surface is being constantly bombarded with high levels of ionizing radiation. And obviously these are not conditions that are very hospitable for life. But we know that Mars hasn't always looked like this. We know that around 3 to 4 billion years ago, Mars had liquid water on the surface. We know this because we have geomorphological evidence, like the presence of ancient river deltas, but also the presence of hydrated minerals on the surface of Mars that we have detected both in situ and in orbit. So this makes astrobiologists very excited because it gives us a potential habitability window between the early Noachian to the mid-Hesperian, which also coincides with the emergence of life on Earth during the early Archean. So this raises a question, which is, if Mars was able to develop life just like the Earth, then what could have potentially happened to it as the planet started to dry out? Well, any superficial bodies of water on Mars would have progressively become more hypersaline with evaporation. And so any life forms that would have inhabited those waters would have to be adapted to high levels of osmotic pressure. And we know those organisms to be halophiles, or salt-loving organisms. So maybe halophiles would have been the potential last survivors. But eventually, these waters would have completely evaporated, leaving behind evaporite minerals. So here is a map that shows the distribution of different hydrated minerals on the Martian surface. And we can see the presence of many different types of evaporites. We have chlorides in yellow, sulfates in pink, some carbonates in green, and overall evaporites are globally distributed on the Martian surface. Most of the chlorides, like sodium chloride, magnesium chloride, and potassium chloride, they were formed during the Noachian, while most sulfates, like magnesium sulfate, sodium sulfate, were deposited mostly during the Hesperian. But why are evaporites so important to astrobiology? Well, as evaporites are forming, they can trap these little pockets of brine, so the hypersaline body of water, in the crystal structure of the evaporites. They can be preserved over geological time. And these tiny pockets of brine, they're called fluid inclusions, as we can see in red over here. And on Earth, fluid inclusions are known to trap organic matter, but also life itself. So here, in these red dots are the autofluorescence of this microbe that I will not try to pronounce. But what is interesting is that fluid inclusions can then act as little lifeboats for prokaryotic life. And on Earth, obviously we have been so scientists were able to isolate and cultivate some microbes that were stuck in fluid inclusions that were formed between the Permian and the Triassic, so a long time ago. And so this leads me to the overarching goal of my PhD project, which is to determine the effect of different Mars-relevant salts on habitability, but also on the preservation of biosignatures in fluid inclusions. So we have two main ways that we can achieve this goal. And they are complementary, such that they are two pieces of the same puzzle or both sides of the same coin, which are the study of analog sites, like Veli was able to talk about before, but also we have lab simulations, and each have their pros and cons. So analog sites are definitely more realistic, but they do add a layer of higher complexity. On the other hand, lab simulations, they are less realistic, but they offer higher simplicity. My PhD is focused on lab simulations, but still I was very fortunate to be able to visit an analog site. Along with Dr. Felipe Gomez from the Center of Astrobiology in Spain and Dr. Velislava Ilieva and Dr. Michael Macey, we went again to the hypersaline lakes of the La Mancha region that Veli has described, and we collected samples from the same sites. And what is cool about these hypersaline lakes is that they are a thalassohaline, meaning that the most abundant salt is not sodium chloride, are any other salts, and in this case they were magnesium and sulfur rich. And among many things that we sampled, we were able to collect some salts both from the subsurface, like this one, which is most likely bluidite, but also others from the surface. And now I'm going to attempt to show a video. So this is all, these videos were all taken from Peña Hueca, only one of the 4 sites. And as you can see, each of these tiny squares, these are halite crystals. So everything that is white on the lake that you're going to see are basically this that you're watching right now, halite sodium chloride crystals. Again, this is Peña Hueca. All of these are halite crystals, which is pretty cool. We can see how abundant they are in the environment. Here you can have a global context of the site, and that's Michael and Veli. Also here, before the video was looking at the lake, and now this is on the borders of the lake, where water has completely evaporated, and you can see, maybe if I do it like this, all of these things are also salt crystals that you can see are super abundant. In the video, I'm gonna pick one up, and you will see on the background, the things that are shining are these crystals, and you will be able to see how abundant they are in the environment. So these crystals were bigger than the halite crystals that I showed you before. They were considerably bigger, and they were super abundant on the edges of the lake. And everything that shines on the background, those are also the same crystal as the one that I am holding right now. All right. But as I said, my PhD is about lab simulations, and to understand how different Martian relevant salts have an impact on habitability, and also how they can preserve differently biosignatures in their fluid inclusions. My project is split into 3 main parts, which are in essence evolution, desiccation, and destruction, and I'm gonna go through each of these stages one by one. So the first part of my PhD is to characterize the evolutionary consequences of exposure to Martian-relevant brines. And I'm going to do this relying on microbial experimental evolution. So experimental evolution is when we use controlled laboratory populations and environments to study the mechanisms of evolution in real time, where selection can act on any and all traits and genes relevant to fitness. This is not a new science. It most likely started with William Henry Dallinger, which was a contemporary of Darwin, and with also his infamous experiment that lasted over 6 years, so from 1880 to 1886. What William Henry Dallinger did was that he went to a local lake and he took some samples and he was able to isolate some protozoans. We are not entirely sure what those protozoans could have been, but still we know what he did in his experiment. So the lake, let's imagine, was at around 10 to 15 degrees Celsius, and he wanted to see if his protozoans could survive high temperatures. So he exposed the protozoans to 70 degrees Celsius. And just a detail, basically in these images, these protozoans, they are meant to signify a population and not an individual, because evolution doesn't happen at the individual level. It happens to a population. And so, as he exposed the protozoans to high temperatures, 70 degrees Celsius, they died. So basically, an abrupt change led to the extinction of this population. So he decided to take a different approach, and that was to gradually increase the temperature over the period of many years. So each time he would increase a fraction of a degree until he eventually reached 70 degrees Celsius. So initially, he put his microbes at 23 degrees Celsius. They already did not like this temperature, but still they could survive. And he let them stay at the temperature for a while before he started to increase the temperature until the microbes, the protozoans, were able to withstand 70 degrees Celsius. So also an interesting thing happened, which was that the protozoans that were now adapted to living at 70 degrees Celsius, they no longer really liked the initial temperature of 10 to 15 degrees Celsius. And this is due to ecological specialization, where genetic adaptation to a single environment often results in rapid fitness gains, but may result in the loss of fitness in other environments. Again, after being exposed to 70 degrees Celsius for a long time, they no longer liked the initial temperature. There is also another very famous experiment, which is called the long-term E. coli experiment, in which the longest running evolution experiment involved 12 populations of Escherichia coli that were founded from the same ancestral strain, and they have been serially propagated in the same glucose-limited medium for over 30 years. So we're talking about more than 60,000 generations of E. coli. So a good thing about microbial experimental evolution is that microbes are able to duplicate very fast. And this is an example of what happens in terms of mutations in just 20,000 generations. So the experiment already goes beyond 60,000, but here in this graph we're just looking at the first 20,000 generations. And here we have the relative abundance of different mutations. And what you can see is that many mutations appear. Some of them, especially the ones that have these dots before them, are able to be fixed, while others are transient. For instance, this one doesn't have a dot. It was not fixed. One thing that I would like you to pay attention is that here at this time point, which is around 2,500 generations, we already had many mutations that appeared. So even in few generations, in comparison to the 60,000 generations, many mutations were already spotted. Beyond point mutations, there are other things that can happen during evolution, which can be deletions, inversions, and so on, insertions. And what I want to do is to use experimental microbial evolution of 2 model halophiles to determine genetic adaptations to 3 Martian relevant salts. So I'm going to be using Haloferex vulcanii from the Archaea domain. Haloferex vulcanii is from the Halobacteria family. It says Halobacteria, but it's an Archaea. And this family is basically one of the families in Archaea that can withstand the highest concentrations of salts. And among them, Haloferex vulcanii is one of the fastest replicating ones. And I will also be using Vibrio nitrogens from the bacteria domain, also a moderate halophile, and it has a very fast replication time, faster than E. coli in optimum conditions, which is obviously beneficial for this type of experiments. And what I'm going to be doing is to expose them to high concentrations of each of these 3 salts individually. I'm going to be using sodium chloride as a benchmark salt, simply because most of the adaptations that we know of from Earth organisms to hypersalinity, they are based on sodium chloride, simply because it's one of the most abundant salts that we have on our planet. But also it's relevant for Mars because it has been detected on the Martian surface and in Nakla meteorites. We are also going to be using magnesium sulfate, one of the most abundant sulfates on Mars, and magnesium chloride, which was also found on Mars, for instance, as biscophyte, but it was also chosen for a strategic reason. When we pair magnesium with sulfate, we have a cosmotropic salt, meaning that magnesium sulfate is actually able to stabilize biomolecules. On the other hand, magnesium chloride has a chaotropic effect, meaning that it destabilizes biomolecules. So this is also an interesting effect to see in evolution. The first step of a microbial experimental evolution experiment is to understand what is the selective pressure that we want to drive evolution. And in my case, that is the tolerance, the high concentration of 3 of the salts that I mentioned: magnesium chloride, magnesium sulfate, and sodium chloride. And so I need to understand what is the maximum concentration. And so I have been doing growth curves in the lab, which are shown here. So we have 3 graphs, and they are all regarding Haloferex vulcanii. These are growth curves. On the y-axis, we have optical density at 600 nanometers over time in hours. And if there is an increase in optical density, that means that the media became turbid, and so cells were growing. For sodium chloride, we already had data in the literature, so we were able to confirm that our strain was working as the wild type. So we can see that the optimum was 2.5 molar of sodium chloride. We also confirmed that at 0.7 molar of sodium chloride there was no growth, and after a long lag phase it was able to grow at 4.5 molar of sodium chloride, which was the maximum we were able to dissolve in the minimum media that we are using. So 4.5 molar is the concentration of sodium chloride that I'm going to be using for my experiments. For magnesium chloride and magnesium sulfate, there wasn't a lot of data, and so what we did was test different concentrations. For magnesium chloride, the maximum we could dissolve in our minimum media was 1.35 molar, and as you can see here in this light green color, Haloferex vulcanii was able to grow. So again, 1.35 molar of magnesium chloride is what we are going to use for the experiments. And then for magnesium sulfate, again, the maximum we were able to dissolve was 1.4 molar, and it was able to also grow at this concentration. Currently can you still see the screen? Mm-hmm. Okay. So basically I have been doing this for Haloferex vulcanii. I'm currently doing the same for Vibrio nitrogens, so that we can begin the fun part, which is the experimental evolution workflow. And as seen in this on the right side of the screen, we start with an ancestor population, and we divide it into the lines that we want to stress and a control. Our control is in essence minimum media with all of these salts in solution, but in their optimum concentrations. We also have a single carbon source, which for haloferrous vulcanii, that is sodium lactate, and for vibronitrogens, that is glucose. And the only thing we do on the parallel lines is that we only increase the concentration of the salt that we want as a stressor. And we let them grow for 1,000 generations, changing the media by serial dilution. And for haloferrous vulcanii, we might be able to reach 1,000 generations, while for vibronitrogens, we can reach 3,000, for instance. And the difference is simply how long it takes for them to replicate. Haloferrous vulcanii, when it's stressed in these conditions, it takes from 16 to 20 hours to duplicate. My PhD is meant to last 3 years, so there's only so many generations I can do in this time. But vibronitrogens is much faster, so we can reach more generations in the time span of my PhD. What we then do is we compare the metagenomes in the beginning and in the end of our experiment, and we try to identify single nucleotide polymorphisms. So these mutations of a single nucleotide that may confer adaptations to the salts that we are using as a stressor. Another interesting thing we can do is to compare the growth rates in the beginning and in the end of our experiments. And in theory, by the end, if they were able to adapt, they should be faster at growing in the conditions that we submitted them to. This is also a way to measure fitness. So I said a lot of things about evolution, and now I would just like to also tell you what I am thinking of doing for the rest of my PhD. And the second objective is to try to define the impacts of brine chemistry and desiccation on the formation of fluid inclusions. So what I'm going to try and do is expand the brines that I'm going to be working with. So going beyond sodium chloride, magnesium sulfate, and magnesium chloride, I want to test many more. And I will want to evaporate them to form evaporites in different ways. I will test how differences in pressures can impact the number and size of fluid inclusions. So if earth pressure, comparing earth pressure and hyperbaric conditions such as 6 millibar, which is on average the current pressure on Mars, but also other pressures that are more relevant to the Noachian to the Hesperian transition. Compare Earth and the Martian atmospheres and see how all of these factors combined impact the size and the number of fluid inclusions, and therefore how well they can trap the target strains that I'm working with. To do this, we are thinking of using the low pressure chamber that we can see in this photo that was developed by Dr. Álvaro del Moral from the Astrobiology OU group. And just to give you an idea of how important the pressure effect here is, these are some images that were taken from Feng et al. 2024. These images on the left side, they refer to ambient pressure, earth pressure, while on the right hand side it's a low pressure, 6 millibar. And we have 3 different salts that are forming, which are halite, so sodium chloride, epsomite, magnesium sulfate, heptahydrate, and gypsum. And we can see that in earth pressure, all of them are able to trap microbes in their fluid inclusions. As for halite in low pressure, it seems to do better, so be able to trap better microbes in their fluid inclusions. Epsomite, it doesn't really feel the difference, while gypsum is not able to make fluid inclusions at low pressure. So this is something that we want to expand on different salts and also study other pressures. Towards the end of my PhD, I also want to evaluate the preservation of biosignatures in fluid inclusions under destructive processes. So I will want to form these evaporites in the presence of perchlorates, but also of radiation, such as UV and gamma rays, potentially. And we want to make a small catalog of biosignatures using Raman, which is relevant for the Perseverance rover, FTIR, which is relevant for Curiosity, and near-infrared spectroscopy. But this is towards the end of my PhD. So I said a lot of things, and the take-home messages are that halophiles could have been the last potential survivors on Mars due to their ability to withstand high osmotic pressures. What is interesting is that they can be trapped within the fluid inclusions of evaporites, which we know are globally distributed on the Martian surface. So my PhD is broken down into 3 main parts, which are evolution, desiccation, and destruction. I'm currently working on the evolution part, using microbial experimental evolution as a tool to identify genetic adaptations to high salinity. What I have been doing is to characterize Halophyrexylkenii to understand what is the maximum concentration it can tolerate of sodium chloride, magnesium chloride, and magnesium sulfate. I'm currently doing this for Vibrio nitrogens so that I can begin the experimental evolution process, which will be long, but hopefully worth it. I would like to thank my supervisors, Dr. Michael Macey and Dr. Vic Pearson from the Open University, as well as Dr. Tarek Majanati from the University of Essex. I would like to thank the Astrobiology OU for hosting me for my PhD, and I would also would like to thank you so much for the opportunity of speaking in your conference, also being an early career researcher, and thank you for your attention. Thank you.
Thank you again, dear Rita. It was really a good presentation. And if it's okay with you, now I am taking the questions from the chat.
Got it.
Okay, first question. How sure can we be that organics found in them aren't contamination from Earth?
So, for instance, you mean the Perseverance rover, in the sense that it is taking the course, and so they may contain some evaporites, and maybe we would like to look for organics in the evaporites that may be found in the cores. That is very important. If we do get to have sample return, that is definitely something that we need to take into consideration. Also, when people try to extract microorganisms from fluid inclusions, from evaporites, there's also a very rigorous process of doing surface sterilization of the crystals. And also an interesting way to do that is to contaminate them on purpose with microbes that you know who they are, and then you do the sterilization techniques, which can include bleach for certain evaporites, and then you try to extract the microbes from the fluid inclusions. And so if you don't find any of the microbes that you contaminated on purpose, then you can make sure that what you are finding is actually something that was trapped in the fluid inclusion of the evaporite, which doesn't have to be a microbe, can also be other types of organics. Yeah.
Thank you. Next question is, do you think these inclusions could be the last habitable places on Mars, or just one of many?
Ooh, interesting. So anywhere in the solar system where we could find evaporites that could potentially have fluid inclusions, we could find life there, maybe, potentially. We are confident that there are many different types of evaporites on Mars. We have seen many of them in orbit, but also in situ with rovers. I don't know about icy moons. I'm a Mars person. I don't know about icy moons. That is a question maybe for someone else in our group. But yes, anywhere you'd find evaporites, in theory, you could trap microbes. Yeah.
Next question is, what future missions could help us study fluid inclusions directly on Mars?
I love this question. So already Perseverance is on Mars and he's taking the course, like I mentioned. It would be great if we could have sample return. So already we have a mission there working for this purpose, not just fluid inclusions, but potentially if they are evaporites, we can bring them back and study them. But as Veli mentioned before, the Rosalind Franklin mission is going to be very exciting. because we are going to also be digging 2 meters in the Martian surface, and most of the evaporites that we have found, they are on the surface. So it would be interesting to see also how they are distributed also in the subsurface. So Rosalind Franklin rover, I hope everything goes well in the 2030s.
Okay. It was really great presentation, dear Rita. And what conditions are needed for fluid inclusions to form and survive so long? What do you think? Thanks a lot.
All right, thank you for the question. This is very interesting. So for fluid inclusions to form, we need to have a hypersaline body of water. It evaporates and it just makes the evaporite, which can be chlorides, can be sulfates, can be carbonates. So scientists have been able to isolate and cultivate microorganisms stuck in the fluid inclusions from the Permian and the Triassic, right? So that is crazy. And there are some theories as to how microbes can survive in those fluid inclusions for so long. There's a proposed state of dormancy, but also if in the fluid inclusion you have more than one microbe stuck, one can eat the other, kind of, but also there's, in hypersaline environments, one of the primary producers, a very famous one, is Dunaliella salina, so an algae. And if you have a fluid inclusion with that algae and a microbe, the microbe can feed on the algae for a long time. So this is a way that they can survive for a long time. Yeah. I hope I answered the question.
Thank you. Next one is, how can fluid inclusions tell us if Mars was ever habitable?
So what I'm trying to do is understand if I can get microbes stuck in fluid inclusions. I'm trying to understand what types of evaporites are the best ones at different pressures to trap microbes and organics. And what is cool of fluid inclusions is that they can shield whatever is stuck in them from radiation and from the elements. It's basically protecting whatever is inside. So if you have organics stuck in the fluid inclusion, microorganisms stuck in the fluid inclusion, they can help to preserve them. And so if we find organics in fluid inclusions, that is a hint that maybe Mars had either the conditions to be habitable, or it was actually habitable. It had life, which would be crazy.
Thank you. What are the main problems when studying fluid inclusions from Mars meteorites?
Good question. So I never worked with meteorites, so I'm not 100% sure. I don't know if temperature can affect in this case. Also, when the meteorites reach the Earth, they have gone through a lot, right? And that can also maybe change things in the structure of evaporites and maybe affect fluid inclusions. I'm not entirely sure in regards to meteorites. But thank you for the question. I love your cat. Sorry. Yes. Thank you.
Thank you. Next one. How do you tell if something in a fluid inclusion is biological or not?
So, if we find organics, it can be either, right? It's very difficult to understand if it's biological or not. What we're going to try to do is make a catalog. So we're going to try to desiccate these brines, make evaporites with microbes, and we're going to see what is actually left behind. So we need to wait a bit for me to discover that.
Okay. May I ask a question about Mars living ecosystem? Is Perseverance explore any fauna or any cytological bacteria at Mars? Do you have any clue?
I'm not entirely sure I maybe understood the question. So Perseverance is getting these cores and is leaving them. So it collects the core and it leaves it where it was. What we need to do now is get them back on Earth. There's only so much that the Perseverance rover can do in situ. In order to see what can be in the cores, we definitely need to bring them back. Also to understand if the organics that we have found, what could be their origin. And also because the resolution of the equipments on the rover is not the same as in an Earth lab. So there are all of these implications. We just definitely need Mars sample return.
Okay. Are there any other extreme microorganisms worth studying besides H. volcani organism?
Yes, there are. Wow, extremophiles are incredible, and we should study them all, in my opinion. The reason we chose Halophorus volcanii for my project is because it's among the Halobacteria family, which are Archaea. It's one of the fastest replicating ones, so it's beneficial for me in terms of the evolution experiments. I have a member of this family that can reach many generations during the course of my PhD. But there's many other halobacteria that are very important in the Archaea domain that we could also use. It's just that they grow slower, but they're very important in hypersaline environments. And yes, in theory, also for the desiccation experiments, we could expand and try to use other microorganisms for sure. Yes.
Okay.
Thank you.
Thank you for this amazing presentation, said Sude. That was the last one, I guess. Not the last one, but we reach our time limits.
Okay, thank you.
I think they may ask their questions to you by mail.
Sure thing.
Thank you again for presentation and answering our questions.
Thank you for having me. Thank you.
Okay, bye-bye.
Ciao, thank you.
Now we have 15-minute breaks. See you again at the third session.
Hello, everyone, and hello, Dr. Michael. How are you?
Very well, thank you. How is everything going?
Oh, thank you so much. And again, hello, everyone, and welcome back, Dr. Michael Macey. My name is Merve Cenge, and I'm 30-year student in the biology department at Gazi University. Maybe you remember last year, again, Michael, this Congress. Thank you so much again. And in this session, we are delighted to welcome Dr. Michael Macey. He has been supporting our events for many years, and we sincerely thank him for joining us again this year. And Dr. Michael works in the field of astrobiology and focuses on life in extreme environments, particularly halopiles and hypersaline systems. He is certainly affiliated with the Open University. And today he will deliver a talk titled Life in Field, Exploring Habitability through the study of halopiles and hypersaline analogs. And again, welcome, Dr. Michael. Thank you for very much for joining us again today.
Yeah, thank you very much. I will try and share my slides.
Okay. If you have any questions or problem, I'm here to help. Enjoy the presentation.
Brilliant, thank you. Okay, if I stop sharing at any point, please do let me know. And yeah, fantastic. Well, thank you very much to the organizers for having me back. It's, yeah, I think really wonderful, this event and the series of events throughout the year that you put on, the opportunity to explore the science of astrobiology specifically in this Congress. So the areas I'm going to cover today can be summed up into 4 broad areas that we're going to walk through. So the value and ecology of studying analog environments, are more of a pulled out view of...
I'm so sorry I'm intervening, but
yeah.
We are just seeing a blue screen. Is it okay?
Okay, can you see? Let me try resharing.
Okay.
Apologies. Okay. Okay, if I do this one. Okay, I don't have permission to share that one. Hang on, bear with. Okay. Window, presentation. Waiting. There we go. Is that working now?
Okay. Yeah, now working. Yeah,
fantastic.
I'm so sorry.
No, thank you for correcting that before I got 10 minutes okay. In.
Stage is yours.
Yeah, brilliant. Thank you very much. Well, I mean, it was just this content slide. So the value and ecology of analogs, simulating Mars using some of these hypersaline environments and saline environments, genomically exploring what these halophiles are capable of in terms of the limits of life and their biosynthetic potential. So these last 2 really touching on brand new research areas that I've been working on since last presenting here. It's also fantastic getting to go after Veli and Rita, one, because I get to be a proud supervisor and see the wonderful science that they presented, but also because they've done all of the great work introducing Mars and some of the concepts surrounding that. So I get to ride off their coattails in that respect. So my research primarily focuses on hypersaline environments, as we've touched on, and the technical definition for hypersalinity is a salt concentration exceeding that of seawater. As you can see from some of the examples here, we've got globally distributed salt flats. So this is the point where there is either a desiccated lake basin or an expansive salt flat that has formed through repeat desiccation. And many of these can have additional extreme parameters, whether that's fluctuations in temperature, pH, or exposure to high levels of ultraviolet radiation. And in the case of many of these hotter countries, which all of the examples shown on this slide are. Now, why are we interested in hypersaline sites? And we're going to first start off with, because the fact that studying extreme environments allows us to try and understand life at its very limits. So shown here graphically, in dark green, we have all known life that's been found with regards to 4 selective parameters, so pH, pressure, salinity, and temperature. And in the lighter green, we have those that aren't known through cultivation-based study, that are the candidatus organisms, the term that we give to microbes when we know their existence through sequencing-based studies. There are no isolated representatives in the lab. And if we look at the total diversity of life on Earth, we see that there are some incredibly extreme representatives of life on Earth that we're yet to cultivate. But if we compare that to the total diversity of environments on Earth, we know that there are in fact some uninhabitable environments on Earth, and maybe some of those are environments in which we just are yet to find inhabitants, but there are some, such as the heart of a volcano, or at the most extreme possibles of pH and temperature, where we wouldn't necessarily expect to find persistent, viable microbial life. And if we then compare that to what we know about Mars and the environments that both currently and have existed on the surface over the course of its geological history, we also gain an understanding that, again, there are some environments that would have been and would still be uninhabitable, such as the radiation bombarded perchlorate salts on its surface, greatly exceeding any known tolerance parameters of life on Earth. But there is an overlap across the geological history of Mars, where there were ambient pressures, temperatures, and predicted circumneutral fluids. And then we have this bridging out into more extreme conditions. So what can we learn by studying analog environments? We've already heard that an analog is an approximation of an environment on another celestial body, whether that be a moon or a planet. And for me, some of the main things that can be taken from this is an exploration of the ecology and the ecological principles and how they apply to and define life at all levels. If you have a single cell within an environment, it's inhabited. If you then have a division of that cell generating 2 cells, you have a population much more than that, and any genetic divergence is generated within that population just through standard genetic drift and mutation. You then have something a little bit more complex. And as these cells interact, you have ecological interactions. And ecological principles we observe in macroecology from animals and plants and organisms that we can see walking around. Many of these same core principles apply to our understanding and ability to characterize microbial diversity and its persistence under extreme conditions. So microenvironments, this means the stratification of environments. Microenvironments, sorry. We have stratification of selection gradients, whether that be chemical or physical, within an environment. So if we think about a hydrothermal vent in the extremes of temperature surrounding it, as you get further away from the opening of the vent, you get cooler temperatures. So this is a stratification of a selection gradient. Now shown here is a Saharan salt flat that I was able to sample with the Ibn Battuta Research Institutes, thanks to funding from Europlanet. And it's a very expansive salt flat. It's actually a very thick salt crust. And when you crack this open, you have multiple layers of very thick halites. And then beneath this, you do then encounter water, unconsolidated minerals. And then if you dig down far enough, you still encounter sedimentary layers. So we've got this gray mud that's fairly synonymous with, if you dig down far enough, you eventually find gray mud in most environments. But if we think about the environments within this environment, we have the top of the salt crust shown here. We have the sub-layers of halites. So there's still this incredible chemical selective pressure, but we don't have the same physical selection pressure of exposure to ultraviolet radiation. We then have a redox gradient across the depth of these profiles, and that's why it's lighter at the top and darker at the bottom. So we get a diverse array of microbial environments within one specific analog. And this has great relevance to our understanding of extraterrestrial sites, where within exploration of Mars, we have caves, and we would expect there to be variation in the physicochemical gradient at the mouth of the cave to the deep surface, again, with the surface of Mars relative to the subsurface, and as Rita explained so well, to the outside of a salt crystal and the internal fluid inclusions. It's also relevant to our understanding of putative biosignatures. So shown here, we have an example of a possible microbe that could exist that's performing sulfur oxidation simultaneous with nitrate reduction. So it's using nitrates, the terminal electron acceptor, and that's how this metabolism is being fueled. Well, in terms of sulfur oxidation, there's this paper that was led by Dr. Arola Moreras-Marti, published in 2022, that is open access. We look at the isotopic signature of sulfur and how it's influenced by different metabolic pathways. So we have sulfur disproportionation, sulfate reduction. We then have phototrophic sulfur oxidation in different forms. And we see that there is this separation. So the dark green, sulfur oxidation, sulfate reduction, sulfur disproportionation. And then we have slightly more complex, as things always get with environments where we detect this metabolism with greater overlap. But this is a chemical signal that the microbes are leaving behind on their environment, that even if tomorrow every single microbe within that site went extinct, this chemical signature would potentially persist. It's also relevant when we think about the formation of minerals in addition to evaporitic minerals. So now we have a different hypothetical microbe that's simply performing nitrate reduction. Now, there is a metabolism that gets a lot of focus in the astrobiology literature called nitrate-dependent ion oxidation, which is the process of nitrate reduction appears to be tied to ion oxidation. So we have ion in the Fe2 state, so the gray, and then going to Fe3+ when nitrate reduction has occurred, so going to the red, rusty state that we would associate with the surface of Mars. And this is also very commonly associated with, as this oxidized ion is forming, you get encrustation of the cells within these ion minerals that are then precipitating out. You get entombment of the cells. This then means entombment of organic biosignatures that, again, in the right conditions, could persist over incredibly long timescales. And if you're interested in looking into nitrate-dependent ion oxidation more, there is an Astrobiology OU alumnus, Dr. Alex Price, who published a review on this as a potential Mars metabolism and performed simulation experiments using some of these microbes. Now, what we did recently was we got a mutant of Salmonella enterica. So this is a medical-related microbe, but it's a denitrifier nonetheless. And this was provided by Dr. Gary Rowley at the University of East Anglia. So it doesn't have the nitrate reductase enzyme required to perform this specific step. So it doesn't produce nitrates. And we grew it alongside the wild type that does have the capability of producing this step. And we also had abiotic controls. So we just simply, instead of adding cells, we added nitrates. And what we observed is in the mutant, if you knock out this enzyme, you knock out the ion oxidation that's happening. So there's no Fe3+ forming. But in the control that had no cells, but just nitrate, the ion was becoming oxidized. Now, this is an interesting result because whilst nitrate-dependent ion oxidation isn't the only pathway, there are some acidophiles that do have dedicated enzymes that directly perform ion oxidation. The mechanism behind it has never been fully known. And what this result would potentially indicate is that in some of the microbes, nitrate-dependent ion oxidation is an artifact of simply nitrate forming, and then the nitrate itself going and interacting with the ion in the chemical fluid. So you have this direct interaction that's not biological. The nitrate is forming because of biological activity, but no further enzyme is getting involved. It's occurring as a direct consequence of the nitrate being formed and then interacting with ion in the system. But this is an interesting result because ion entombment by cells is still potentially a viable putative biosignature and is perhaps a little bit more pervasive within microbial diversity than we initially thought, because lots of microbes are capable of this first metabolic step of denitrification, converting nitrate to nitrite. So it's possible that with it being tied simply to nitrate formation, that entombment of cells in iron could be a much more widespread occurrence than we would have previously assumed if this had been a process that was instead dependent on the presence of a specific enzyme or biological interaction. Studying analogues can also tell us a lot about metabolic viability and understanding metabolisms fueling microbial communities. So shown here, we have a redox tower. So at the top, we've got oxygen, if you have oxygen as your terminal electron acceptor, as we do in our cells, it's incredibly energy yielding. And then as you go down the tower, you get less energy. So we've got the nitrogenous species here, nitrate and nitrite. We've got ions, we've got sulfates. So as we then look at the different microbes and metabolisms that are possible to occupy an environment in a chemical system that we're studying, and then we compare this to what we actually observe and the genetic repertoire of metabolisms that they possess, this can give us an understanding of, okay, they're viable in this specific chemical system, what may potentially have been viable in former Martian chemical environments. And this connects very strongly with the expansive research that Veli has done, Dr. Velislava Ilieva, in terms of her characterization of the Spanish field sites and the direct chemical relevance to former Martian aqueous environments. And then also the ecological principles and their influence on viability and community assembly. So we've touched on ecology a little bit here, but we also have a complete understanding of the ecosystem. So here's a fairly classical example of an ecosystem, the marine ecosystem, whereby we know that we have interactions between algae, diatoms, fish, and other trophic levels within the ocean. So we have a system comprised of the living and the non-living components of an area. So we also have chemical interactions with the water, with the atmosphere, and with energy input from the sun. But we also have microbial ecosystems where we can't discount interactions and associations between the living and non-living components of a system. So if we think about hypersaline environments, there's one in particular we studied in the Arctic known as Color Peak, and this was published in a paper in 2020, where the community was dominated by sulfur oxidizing bacteria that were fixing carbon dioxide. And this is a carbon poor environment. So very likely these organisms are interacting with the others through the supply of carbon, either leaking or purposely releasing carbon to the environment to modify it, and also simply through dying. These cells are growing, they're dying, and the organic carbon that they leave behind represents an abundant potential carbon source for the non-carbon dioxide fixing members of this community. So we have interactions, we have trophic levels within a microbial ecosystem in the same way that we do within non-microbial ecosystems. And one key concept in ecology is the keystone species. So this is the keystone of the arch. If you remove the arch keystone, the arch will fall down. So this is an organism that has a large effect on its natural environment. If we go back to our same microbial ecosystem within Color Peak, very likely the carbon dioxide fixing microbes that are acting as a carbon pump to the other microbes in this ecosystem are very much the keystone species or keystone function, to the extent to which we can ascribe species identity to microbes. If we were to remove them, it is a question as to would they have a small or large effect on the natural environment. Given their potential associations, we would predict that this would be a large and significant effect. So what can we learn by studying analogs? Well, one key thing we can do is generate hypotheses. So why do we then study multiple analogs? Now, working with a network of international collaborators, I've been very lucky to be able to work with samples from a diverse array of hypersaline environments, and I've also been fortunate enough to visit some of these alongside my collaborators. But why are we studying multiple if we can develop so much data just from the study of one? And there are two key reasons for this. And it effectively boils down to both derived from the fact that no analogs are identical. Now, shown here is a 3D representation of fitness. The higher on the peak, the more fit you are relative to your environment. Minor variations in chemical condition will change where these different peaks will be. Being fitter in a system that has slightly more sulfur in is different from being fitter in a system that has slightly more iron. And this can have major impacts on the viability of distinct metabolisms. Maybe in some methane-producing microbes are excluded, but a simple chemical fluctuation, that's not what occurs. And by studying multiple analogs, we can gain a broader understanding with regards to the viability of distinct metabolisms, the chemical signatures that they leave behind in these environments. And from this, we develop a greater understanding of potential viable metabolisms under former Martian chemical systems. And also because as we've touched on, analogues are an approximation of an approximation. We have an understanding of the surface of Mars from both ground-based missions, the diverse rovers and landers that have taken chemical and physical measurements of the surface, and also from orbital studies and long-range studies using telescopic equipment from Earth. But we still don't have absolute certainty of environments in the same way that we do on Earth, where we're blessed with being able to physically go to a site, collect samples and study them with much higher grade analytical equipment, because we're not constrained of having to get it to another planet first. But also an approximation because Earth has oxygen in its atmosphere and has for a very long time. And even anoxic ecosystems encounter chemicals that exist as a consequence of the presence of this oxygen. So multiple analogues allow us to better explore this analog space. So if in grey we have our proposed understanding of what these former Martian waters would have been like, we have in the darker circles the chemical approximation that the different analogues give us. Some are closer than others, but by studying multiple, we can get a better exploration of this analog space. So we can then push this further. If we're arguing that the microbes within these hypersaline ecosystems and saline ecosystems are viable under proposed Martian former chemical environments, are they viable in a simulated Martian chemistry? So to test this, we used mud from the Colne Estuary. So the Colne Estuary of the River Colne runs through Essex, and it's a saline ecosystem. It's not hypersaline, but we used it to perform some initial tests because it's very well characterized in terms of its microbial community, and it has many metabolic guilds relevant to our understanding of Martian environment. So sulfate reducers, iron reducers, and methanogens have all at one point or another been proposed as relevant as viable metabolisms under proposed former Martian conditions. So what we did was we took this mud from the Colne Estuary, here's its microbial profile here, and we put it into a simulated Martian fluid chemistry derived from the current exploration site of the Curiosity rover, so Gale Crater. So the fluid chemistry was directly derived from the chemistry of that site. But we have the problem of any nutrients present in the mud would have been present here. And if we perform a 1 in 100 dilution, which we did to transfer it across, we still have some micronutrients from the mud. And in fact, to be positive that there was no transfer of these estuarine-derived nutrients, we perform multiple subcultures. And these are the microbial communities at the endpoint for each of these transfers. And we can see that the microbial diversity shifts. Some microbial groups initially increase in abundance and are then lost, and some microbial groups increase in abundance and persist, shown by the black and the purple bars. Intriguingly, methane production was lost after stage 3. So at this specific point, the methanogens could no longer persist. So methane we were seeing was definitely as a direct consequence of mud-based nutrients as opposed to the simulated Martian chemistry. So this was our endpoint community, and we found that it was dominated by sulfate reducers and acetogens, so microbes that either produce reduced sulfur species as the endpoint or produce acetate, a carbonaceous species, as their metabolic endpoint. And the results of this study are shown here. So the question for are microbes viable in the simulated chemistry was yes, even after we ensured that all of the original nutrients from the natural environment they were derived from were eliminated. Yes, they were viable in the simulated chemistry, but do they remain viable in divergent simulated chemistries? Are there differences in the metabolic viability as a consequence of these differences between the chemical environments? Would we expect there to be differences in microbiology? So terrestrial environments with similar chemistries contain similar microbiomes, and this is something persistent we see. There is selection for specific taxa in metabolism. So in the same way that Veli observed similar metabolic diversity with some fluctuations between her sites, anoxic and oxic sediments under hypersaline conditions, we would expect selection that resulted in an overlap of taxonomic and metabolic diversity. So shown here, we have 2 salterns, which are desiccated salt flats, one in Puerto Rico, one in Spain, and we can see incredible overlap in the microbial diversity. If we add alkaline pH as an additional selected parameter, we have here microbial diversity between Gar Lake in Egypt and Bitter Lake in Russia, again, massively geographically separated, but with significant overlap in the microbial diversity. and grassland soil, adding in a non-extreme representative here, we see comparison a grassland field in Germany to one in the east of England. We again have significant overlap. And if we then compare between them, we see that there is major shifts between the different representatives of these environment types, between the saltons, the soda lakes, and the grassland soils. So yes, we would potentially expect differences in the microbiomes of sites with incredibly distinct chemistries. So for the purpose of our exploration, what we did was we looked at the chemistry of rock nests, so the specific site in Gale Crater that we performed the initial experiments with, but then we also used selection on hematite slope, which is a more Fe3+ rich environment on the Martian surface, and Pastel Robles that was characterized by the Phoenix Lambda and is a much more sulfur-rich environment. So we set up our experiment using this enriched microbiome that can grow in the absence of any external nutrients from the initial environment. They're growing solely on the supplied chemical Martian environment. And we performed an array of microbial and geochemical analysis in order to understand, okay, are these viable and how do they vary? So this was the experimental setup, and as ever, the devil is in the details. So the abiotic controls were completely necessary to run alongside. So these are identical. The only difference is that there is no cell supplied to these systems. So do we observe variations between the chemistries in terms of microbiology? In terms of observations of growth, yes. So in black, we have rock nest. In red, we have hematite slope. And in blue, we have Pastel Robles. So we do see, yes, there's variations in the growth pattern. It's very consistent between replicates, even though we provided a mixed community of microbes, and it indicates there is variable lag phases, growth phases, death phases between these chemistries. And we thought, okay, fantastic, we're going to sequence all of these. We're going to compare between replicates, and I wonder how this reflects in terms of the diversity. And there was zero difference in the microbial diversity. We see the same three genera dominating all of the fluid chemistry. So we've got the inoculum that was enriched previously on our Martian fluids. We see that there is 2 sulfate reducers shown in red and acetobacterium shown in black. They vary slightly in ratios, definitely, but overall we observe the same 3 genera and elimination of any other bacteria that were present in the inoculum. And we thought that this was a bit of a surprising result, and it must simply be that the dominant selective regime is for sulfate reduction and acetogenesis, and any subsequent chemical variation between them isn't sufficient for that chemical selection for specific metabolic viabilities to not be overwhelmed and result in distinct microbial communities. But of course, we perform this with a highly selected and reduced diversity inoculum. So we performed it with the stage 7 microbiome, but of course we have samples from all of the previous stages. So what we did was we went back to when the microbial community was much more diverse, when methane production was still present, and we performed exactly the same experiment. So the question is then, do we observe differences in the enriched microbiome? Taxonomically, yes. So suddenly, immediately, we see differences. So we see the same red bars in the rock nest chemistry, the same black bars in the rock nest chemistry, but we're starting to see yellows and oranges representing distinct taxonomic groups. That's the key takeaway from this graph, is in the other enrichments, we did see these different colored bars that weren't present in the previous graph. But they're all sulfate reducers. They are very much all sulfate reducers. So we see that whilst the taxonomic diversity varied, the metabolisms were identical. So this environment very much selects. When the specific array of chemical simulated environments from the Martian former aqueous history were used to select against a few different arrays, microbial combinations, we always saw sulfate reduction. We always saw acetogenesis. We performed these experiments with a different environmental inoculum, and those are written up and shared in this other open access study, where we instead used a hypersaline inoculum, and we found effectively exactly the same result. So what's the significance of seeing this same shared metabolic enrichment between the sites? Well, if we think about how communities are assembled, everything has gone through a filter of selection. So we started with a more diverse inoculum, and then we ended up with these same 3 things. So same colors, red, black, acetogenic bacteria, sulfate reducing bacteria. And these are the metabolisms that they're using. So converting sulfates to reduced sulfur species and also producing acetate. This has major consequences on how they'll influence their environments, because if we have exactly the same metabolisms occurring in different chemical systems, that should affect the chemistry of these environments in the same way. In terms of analysis of the chemistries, that's exactly what we found. So sulfide and sulfate were both eliminated from the fluid chemistry. So we proposed that is because sulfate reduction was converting the sulfate to sulfide. Sulfide was binding to the metals, and it was then removing them from the fluid chemistry. And this is paired with the fact that we see a reduction in iron chemistry and aluminium. So we're getting this formation of sulfidic minerals. And using electron microscopy confirmed these common shifts between fluid chemistry and distinct changes that were detectable between the abiotic and the biotic vial. So fantastic. Initially, we were very happy with this result because it gives us an indication as to maybe potential chemical categories for the evolution of systems, and maybe these are potential categories of biosignature. However, in terms of the bad, we also performed thermochemical modeling, which allows us to assess from the abiotic fluids if these aged over geological timescales, what would be the resulting fluid variation that we observed in the absence of any biology. And it's a different story of what we then see. So in the abiotic fluid chemistries, if we model these over geological time, exactly the same pattern happens. If you have a reducing fluid, sulfate will gradually transition to sulfide, and the sulfide will gradually bind to the metal ions. And we do see a slight reduction relative to the starting chemistry in the abiotic test groups, indicating that process was starting, and given enough time, we would see that continuing. So biology is very good at accelerating processes. It's very bad at performing processes exclusive to biology. So the significance of this result is it does show us that if we have the filter of selection, as we observed in our simulated Martian chemical environments, it does have a direct result on the endpoint community that we observe. We saw consistent enrichment for these specific taxonomic and metabolic groups. So this wasn't simply random variation. This was selection for the viability of sulfate reduction. But it also reinforces the complexity of trying to identify biosignatures solely through chemical and mineralogical identification alone, and it potentially requires more in-depth analysis, as Rita was touching on, the importance of Mars sample return and much more sensitive analytical techniques. We also have the question of, would a distinct microbial community have an identical chemical impact on the environment? So if we went back and did this again with yet another inoculum, we do in fact observe similar chemical selection for specific microbial taxa. So that was the key takeaway that we took from this work. Now we're going to move on to the question of, why do we study halophiles? And the second point that I brought up was extremophiles possess adaptations to the extreme selection pressures that they encounter in their high salt environment. So shown here, we have a figure, and it shows broadly the two different adaptive strategies common to halophiles. So we have the salt-in strategy, where you have the transport of salt ions into the cell, and then simply have a proteome that's acidified and capable of persisting in spite of the high salt chemistry, low pH, and effectively just harsh internal cellular chemistry. The salt-out strategy is you produce these organic osmolytic compounds like glycine betaine, DMSP. These balance the internal osmotic charge, and it means that the water doesn't just transport out of your cell and kill the cell. So we typically observe both in Halophila archaea and bacteria, a combination of these two adaptive strategies. But we see further adaptations in some of the Candidatus microbial taxa that remain uncultivated. So what I'm going to touch on here is a side project that I've been working on that very much has turned into a bit of a passion project, exploring the genomic evolution and adaptation of a remaining uncultivated extremophilic microbial taxon. So in the study by Vavarakis et al. In 2018, they went to 4 separate soda lakes in the Russian steppes, and they generated 871 metagenome assembled genomes. So packaging that sequencing data back into proposed genomes. And this study really laid the foundation for a lot of understanding of the uncultivated microbial diversity we observe in halophilic environments to this date. Now, of these 871 mags, one was T1Z10-126. -126 because it was the 126th from that specific environment, T1 because it was from Lake Tanatar. Now, this was a genome that was 2.2 megabases, 121 contigs, 48% GC content, good completeness, 0% contamination, and it was classified as a bacterium. And the databases couldn't classify it further than that. Now, in addition to this one, there was also an extra one from Cock Lake in the same Russian steppes that clustered to the same group. So we have 2 representatives from this study of this microbial group. 3 studies since 2018 have explicitly reported the detection of this microbial group, and this has been in a Polish salt mine. This has also been in salt flats in Ethiopia, so specifically the Logrot region of the Danakil Depression, and in the Odil salt marshes in Spain. So this is a very recent publication that expanded the known diversity of this microbial taxon. So in this study here, so Gutierrez Garcia et al. 2024, they proposed the name Candidatus Sausurobacterota, so replacing the affectionately named T1Z10-126. And this is a big movement within microbiology to provide names to some of these persistent Candidatus taxa. Just because they're uncultivated doesn't mean that having a name isn't useful for the purpose of furthering this study. And as well as providing a proposed name, this study also generated 2 representatives of this proposed phylum. So that brings the total to 6. So we then performed a broader search of metagenomes around the world using Sandpiper that lets you explore the total known diversity of all metagenomes with specific taxonomic markers. So we genuinely searched phylum T1Z10-126. It screened 220 metagenomes, and it showed that it was present in globally distributed environments at 0.01 to 1.26% relative abundance. So it is found globally in a diverse array of environments, but it is never seemingly a major player in terms of abundance. Doesn't mean it's not having a major impact on its environment, but never numerically abundant relative to everything else. Now, what's really cool with where it detected these is previously we've detected it in hypersaline environments. Those studies that reported detection were from saline and hypersaline environments. Sandpiper also identified some additional moderate environments. So the Odil salt marshes study, the Odil salt marshes are saline, they're not hypersaline. So there's a broader physiological tolerance in this proposed phylum than just hypersalinity. It was also detected in anthropogenic and subsurface environments, including oil wells in the United States of America, as well as some xeric soils, so incredibly dried soils, but there's no salt there. So it indicates a survival tolerance of desiccation as well as high salt exposure. And then also there is a metagenome of paleofeces, so desiccated feces that's been preserved, and it was detected in this. So what we did was we selected an array of these metagenomes to expand the geographical range and potential niches that TSED has been shown to occupy. And from this, we were able to assemble 26 non-redundant genomes spanning 12 countries and 10 distinct environmental categories. So from oil wells to salt flats to the salt marshes. So a broader genetic array than we were initially starting with. If we look at comparisons between all the different genomes that we assembled, we see incredible high genetic diversity. So this is a phylum. If we think of any other phylum of bacterium, we would again expect there to be this broad genetic diversity. But what's really interesting is we observe an overall broad pattern of clustering, where the blues aren't quite as dark when we're looking between relatives. We also have some pockets of higher relatedness where it's even closer towards green and yellow, indicating more similar genomes. And this isn't something tied to geography. We see that some of the genomes assembled from Ethiopia cluster very closely to the genome assembled from Poland. So this indicates that perhaps these are distinct lineages, maybe distinct families or genera that have been distributed around the world. So we need to get a further level of analysis than only thinking of this as just one collective phylum, but also the broader breakdown of taxonomic groups. Again, if we then looked at the phylogeny, so we have these same groups that were just shown on the average amino acid identity plot, just now here shown on a phylogenetic tree assembled from conserved housekeeping genes. It again shows the same two broad clades of genomes. So this is something persisting at the overall genome comparison level and comparison of taxonomic marker genes. Now, none of these microbes are in isolation, but we can make inferences about their physiologies. We already have made assumptions with regards to the fact that they're very saline tolerant, but using a program known as Genome Spot, it looks at the average amino acid frequency within the genomes, and it can make inferences about salinity optima. And it found that these are all microbes that can tolerate salt, aligning with the broad geographical distribution that we're observing with them. And this isn't something that varied in terms of their phylogenetic distribution. So the purple box and the blue box didn't really vary from one another in terms of their salt tolerance. But if we look to pH tolerance, we see a significant variation, with the upper clade having a greater tendency towards occupying alkaline and having tolerance of alkaline pHs, and the lower clade having a greater tendency towards tolerance of lower pHs and bordering on circumneutral to acidic. There's also a greater tendency towards oxygen tolerance in the lower clade. Now, if we look at the core genome, if we boil all of these down to what's shared between all of them, we identify a core of 345 gene families. Now, this includes the complete pathways for your central carbon metabolism. So that if you think about the pathways you're studying as part of your courses, we've got the classics, glycolysis, the TCA cycle. We also have enzymes associated with the breakdown of complex materials such as starch. We also see synthesis of all of the essential amino acids as well as nucleotides. So these are the fundamentals that cells require to be capable of heterotrophic growth and not be dependent on an external supply of amino acids and nucleotides to survive. So in a way, maybe it's not surprising that we detect these within the core of the phylum. But we also observe the universal presence of shock proteins in order to, these are associated with being able to better respond to stresses within the cell. Sodium and hydrogen antiporters associated with pH and salinity tolerance, and the potassium uptake system that I mentioned in the very side about the adaptations that we don't observe elsewhere on the tree of life. So this is present in the root of this taxonomic group. Now, if we then perform ancestral genome reconstruction, so we have this core node here. What we're doing is we're walking along the branches and we go, okay, if we look at these end members, we know their genetic composition. But what is shared here, and what can we infer about what was present at this node? What's shared here, what can we infer about this node? So on and so forth. And if we walk backwards, analysis of the ancestral genome indicates that this node, this ancestor for all of these microbes, was an anaerobe. And this is based largely on the complete absence of anything associated with oxygen tolerance within the genome and the conserved presence of an ATPase that we only observe in anaerobes. Halophily. So it was halophilic, and this is again derived from the presence of the potassium uptake system, and an alkalophile. And this is based on the consistent presence of the sodium hydrogen antiporter, as well as the fact that the branches that are most closely related to this ancestor node are associated with alkalinity, physiological predicted tolerance, and detection in alkaline environments. And as the ancestral strain underwent expansion into these diverse environments, the expansion of the genome was primarily driven by variation in the metabolism and respiratory pathways. If we look at the mobile genetic elements in the genome, because of course bacteria don't evolve in a vacuum, there are viruses, there are transposons, there are transmissible genetic elements, we see incredibly high evidence of adaptive horizontal gene transfer occurring multiple times across the expansion of this proposed extremophilic, halophilic phylum. This is something that gives us a greater understanding with regards to habitability of extreme environments on Earth. Halophiles also have applicability to our understanding of habitability of extraterrestrial chemical environments, but they're incredibly diverse across Earth. So seeing this diversity across such an array of environments and seeing common and divergent adaptive strategies within the expansion and evolution of this phylum gives us a greater insight into the processes of evolution itself. So one thing that we observe is specific gene families occur multiple times across the tree. Now, what that means is this strain may have one specific pathway that's also present here and is also present here, with results indicating that it's not a trait that was lost by the other strains, but a trait that's been acquired independently multiple times over the expansion of this strain into novel environments. So if we break down the adaptations, our purple and blue boxes from the ancestor, what do we see? Well, we see that clade A appears to be specialized for anaerobic and alkalophilic niches, whereas clade B has adaptations to oxic conditions. Major differences are the type of ATPase. As I mentioned, the A-type ATPase is the type that's in the ancestor, and this is associated with anaerobic metabolism. And this is lost in members of clade B, where we instead see the F-type ATPase, as well as cytochromes and other machinery required to tolerate oxygen, which, if you do not have those components, is an incredibly toxic compound. We also see an intriguing difference in the osmolytes that they produce. So we see that in clade A, there's genetic capabilities for producing glycine betaine, whereas we have alternate osmolytes produced in clade B, trehalose. Now, they broadly function the same way, but they have different genes required to produce them. And intriguingly, for some reason, at that branching point, they seem to have gone in different ways. We also see variation in the metabolic adaptations between these different clades. We see genes associated with using carbon monoxide exclusively in members of clade A, as well as metal detoxification systems, as well as additional acquisition of materials required to tolerate greater alkaline pHs, meaning that it has further specialized in this specific clade. Whereas in the sister clade, we see none of that, but we do see acquisition of genetic machinery required to interact with nitrogenous species, so using nitrates and nitrous oxide, as well as the greater acquisition of phosphates, so implying that there is a more efficient system for acquiring phosphates within these microbes. So really, the study of this Candidatus phylum, and even though we only have 26 mags allowing us to explore this genetic diversity, it's fascinating to see these divergent adaptive processes, because these both are clades that occupy extreme environments, but they have chosen very different strategies, or evolution has selected for divergent strategies from a common ancestor. And this gives us a greater understanding with regards to the metabolic viability of these different pathways within the distinct systems in which we detect them, with clade B being excluded from alkaline environments and clade A members predominantly being excluded from oxic environments where there is the presence of oxygen. Now, going back to conclude on a project that we've been working on at the Open University, we're also interested in hypersaline sites because they represent a potential reservoir of novel antimicrobials. So there is a global antimicrobial crisis. Resistance is incredibly widespread and is expected to become an increasing problem as we go into the future. Shown here are just two examples of halophiles, specifically actinobacteria, that were isolated by other research groups from the Atacama deserts that were shown and confirmed to produce metabolites with novel antimicrobial activity pathways. And it's as a consequence of two key things. One, the adaptations that we don't really observe elsewhere on the tree of life. This also includes metabolites. And we have to remember that microbes produce compounds with antimicrobial effect, either as a side effect of the intended purpose, so metal acquisition, and it happens to kill other cells, or to intentionally kill competitors so they can better occupy and acquire nutrients for themselves. Or simply because hypersaline environments have been overlooked in terms of potential applications of their metabolites in terms of novel antimicrobial and antifungal compounds. So we're going, for me, much closer to home than the sites we've touched on previously. We're going to Droitwich Spa. So Droitwich is in the county of Worcestershire. and it's just outside of Birmingham. So it's about 22 miles, 35 kilometers southwest of Birmingham. I got permission to use the town's insignia, which apparently is still, misuse is still punishable by execution by beheading. So thankfully the local council gave me permission to use this. And salt has influenced the entire history of this town. It's even in the town's slogan, Sau sapit omina, salt flavors all. So Droitwich is one of many locations in the UK that's located above a block of salt, specifically in this case Triassic salt. So there was periodic flooding and drying of seawater 200 million years ago, and this built up a layer of rock salt. And this was then buried under layers of clay that was left close to the surface of Droitwich. And this became something that influenced the history of the town and became what the town was known for. So there's archaeological evidence of salt harvesting in Droitwich, or what would eventually become Droitwich, as far back as the Iron Ages. And there are brine pumps, Roman brine pumps that they found within what the Romans called the town of Salinae that eventually became Droitwich proper. And the Romans actually used the salt from Droitwich to pay their soldiers because it was of such high value to them and of such high chemical purity. Now, salt harvesting continued over the centuries. We have something closer to the modern day shown here in the top. And there was also a salt bath that existed in one form or another from 1836 through to 2008, and is currently in the process of being rebuilt after a couple of decades of closure. And people went from all around the world to Droitwich. It's the reason it was given the spa title was because it had these waters that of proposed therapeutic benefits. And if you go back into the literature of what was explored at the time, you also come across some quite strange papers from the 1800s. So I just wanted to highlight this one specifically, where this doctor is proposing that you could even drink the water from Droitwich because it's so chemically pure. But people, for some reason, don't do this. And of course, the reason they don't do this is because it's salt-saturated water, which probably wouldn't make you feel any better. But it was proposed as a potential curative for some medical conditions at the time. So the aims of this specific work were to look at what was the chemistry, what were the microbes, and do they do anything interesting in terms of their metabolites? So there is currently access to the Droitwich brines from one specific point. This is a historic well that was restored by Churchfield Saltworks, which is a private company. What they do is they draw up the brines, they leave it in these large basins to dry out, and then they sell the salt crystals. So this is similar to salt works around the world. You see similar things in the north of Brazil on a much larger scale. And this is also, other than the assistance of a pump to bring the water up that's powered by an engine, very similar to processes that have occurred throughout human history with the harvesting of salt. So this is actually footage of our sampling, very scientific, into our plastic bucket, which we then decanted into glass bottles for storage. So what was the chemistry? I am fairly consistently on the lookout for extreme environments within the United Kingdom, as well as working with collaborators to explore extreme environments outside of the UK. But it's fascinating because the brines are salt saturated. The density, just the simple weigh a mil of it, it's 1.2 grams. And that's exactly the same density as a 25% salt saturated solution. And if we look at the exact chemical breakdown, it's dominated by sodium. And we also see persistence of other salts, as well as the presence of some trace elements. What was also very fun was, because there are so many historical references to the purity of the salt, it meant that there must have previously been chemical measurements, and we were able to track this image down. This was not preserved in any online repository. This is the image of somebody trying to sell the book on eBay, but we were able to get the chemistry of the water through this old school technique that they used of, they would dry out an imperial gallon and then count the individual salt grains. And comparing the modern to the historical chemistries, we see that there is broad agreement. Sodium is the dominant salt, calcium is also present, sulfur is also present, as well as magnesium. We don't really detect the trace elements. This is presumably because that previous technique wasn't very sensitive, but it's also interesting to think about is this potentially also reflecting some chemical fluctuation that's occurred over the last 200 years that we're now detecting, or is it just that the ICP is a more chemically sensitive technique? We went with an isolation approach, so we could use these for screening, and we enriched the microbes on marine broth. So this is a very nutrient-rich medium from which we got multiple strains of Halomonaceae, as well as a few other different strains. And from the Halomonaceae, we selected a specific strain of Vreelandella to study further. And we were able to really dig down into its genome to explore genomically, how is it surviving in the chemical selective environment of this brine? Now, when we looked at its 16S gene, so its barcode marker gene, we found that it was very closely related to Neptuniae, a strain that was isolated from the ocean. And that's already of interest because that means it's adapted to salt, but seawater is significantly less salt enriched than the joint-rich brines. So we were curious with regards to what variation we would see between it and its sister species. Looking at its genome, it has the genes for the central carbohydrate metabolic pathways, some nitrogen interactive pathways, so it can make nitrite, and that's as far as it gets through the pathway. It also has an active virus in its genome. So we're able to predict that this virus in its genome was intact, and exposing the strain itself to ultraviolet light resulted in induction of the phage. We literally saw clearances in the plate where the phage became active and lysed itself and killed the host cells that it was within. This gives a really interesting insight into, again, the ecology of these brines, because it shows that viruses are very relevant to the persistence and selection of these different strains and their genomic evolution. Now, when we look at the total genetic contents of Vreelandella, this cluster here, these strains have got the largest genomes out of every member of this genus, apart from our pet strain here, the Vreelandella strain that we got from Droitwich. There are 711 genes that are absent in our strain that are present in all of the marine relatives. So it's lost genes relating to carbon scavenging, iron acquisition, and B12 synthesis and transport. And it instead has genes more relating to metal, salt, and viral resistance, indicating that this is the specific genomic reduction and genomic expansion that it's undergone as a result of the selective regime of its transitioning to a more hypersaline environment since sharing a common ancestor with these marine relatives. So iron acquisition and carbon scavenging benefit the marine strains, but these were less important to maintain in the genome in the brines. What metabolites did it produce? Well, one stood out immediately. There's a chemical called potashellin that's identified in this paper, and it's a siderophore, so it binds to iron, but it's also been shown to have some antimicrobial activity. We also identified this compound that showed similarity to an antifungal. If we look at where these genes are distributed, potashellin encoding genes are effectively spread to the wind within this genus. They're very sporadically present. But if we look at where the antifungal beta-lactone compound is present, it's only in our strain and the very close marine relatives. So our strain is one of the only ones that has both genetic clusters. This is a significant result because when we screened it against MRSA or MRSA or methicillin-resistant Staphylococcus aureus, as well as other members of the escape pathogens, the most prevalent antimicrobial resistant hospital-associated pathogens, we see this pattern of clearing around the growth of our strain. That means it's producing something that is killing these pathogens. Now, could it be the potashellin? Could it be the proposed antifungal or chemically similar to an antifungal compound? Is it a combined additive effect? We don't know, but we do have an understanding that it's got the genetic potential to produce these compounds, and it will be producing them for a reason, meaning that within its environment, it potentially has the genetic capacity to produce compounds that are killing other halophiles and allowing it to be one of the most abundant members within the Droitwich brines. So in summary, hypersaline environments allow us to develop hypotheses on the limits of life and habitability. And that's not just the habitability of extraterrestrial sites, although it has direct relevance to it, but also the habitability of the world around us. By studying halophiles, we can gain an understanding of the study of evolutionary history itself, and halophiles very crucially represent a potential novel source of antibiotics, as well as other metabolites with important bioindustrial applications. With that, I'd like to take a second to thank collaborators. So Dr. Mara Leitzi, Dr. John T. Munich, and Dr. Veli Ilieva all worked on the antimicrobial work. Daniel and Veli both worked on the TSED project. And Councillor Bob Brooks is a local councillor to Droitwich who really championed our being able to access the brines and facilitated all of that. And of course, microbes don't exist in a vacuum, and neither does science. And I've been very fortunate to work with a lot of international collaborators through my study of some of these environments. So again, thank you very much to the organizers of this event. And I will stop sharing there. I'm happy to take questions.
Thank you so much, Dr. Michael, Professor Michael. It was really a great presentation. Now we have a few questions now. If you're ready, I can go through them one by one.
Yep, sounds great.
So first the questions. Yes, Sansato says, what are the current species of animals that are capable of surviving these resurgent areas?
So Dr. Ben Tatton, who's a graduate from our research group, he always comments that he thinks flamingos should be studied as an example of extraterrestrial life, for, you know, proposed equivalents for, because if we go to a lot of these sites around the world and the microbes are at their very limits of life, and there's just flamingos that are perfectly happy there. In terms of other animals within the system, you do get protozoa, you do get microscopic animals, and then you do have ecosystems. So the lagunas in Spain are very well known for their bird species. That's one of the reasons they're protected sites now. It's because they have many endemic bird species for Spain and for Europe. So you do see animals in some of them, whereas in others, like catch in northern India and the Droitwich brines, we don't really observe any types of animal, and that's potentially just a consequence of the nature of those systems.
Thank you for that great answer, Michael, Dr. Michael. And another question, Zeynep says, what is the most extreme salt environment where life has been found?
That's a great question, and it ties very strongly to Rita's work and the comments that she made about the complexity of studying fluid inclusion. So if we think about how fluid inclusions are formed, where we have this desiccation occurring in the formation of evaporitic minerals, I would say that it's those microfluid inclusions, because you have a salt saturated solution, that's why the evaporites are forming, and then it's concentrated even further. And some of the rocks that I believe have been found by the Perseverance rover, for them to have formed, the chemistry of that site must have been not just as hypersaline as on Earth, but even further and with a great abundance of other salts like perchlorates. So I think it's less a case of one geographical environment and more a case of the concentrated pools and microenvironments that occur within them.
Thank you so much again. And other questions, especially that was actually a question I was curious about as well. So Dennis says, is the evolution of extremophiles found at the Mars different from the evolution of others being in the Earth in case of their metabolic pathways?
So that's a great question. So there obviously has still been no detection of life on Mars, and it's an intriguing possibility because it brings up the broader questions of would it still be viable, even though loss of atmosphere, bombardment of radiation, and would it have been a second independent origin of life event, or would it have been transmission from Mars, because that's broadly the way meteorites travel. It would have been Mars to Earth. I think in terms of metabolism, the reason that it's valid to understand the metabolic pathways that extremophiles use on Earth is because there is a finite number of electron donor pairs. So whilst it may not be, if it was a second completely distinct origin of life event, if it ever occurred, it wouldn't be exactly the same systems, but if there was energy, which as far as we understand all life needs, you would expect there to be that redox gradient. So we would expect there to be overlap in these specific, if not metabolisms how we would observe them, but exchanges of electrons and that redox potential gradient. You were muted, but I see the question is, do you think hypersaline environments are underestimated? I think, to be fair, I think they are probably estimated accurately. There are many different research groups that study diverse hypersaline environments around the world. I think what's also really interesting as a microbial ecologist is revisiting sites that have previously been characterized, because if we study the diversity of animals or plants within a system, we wouldn't do a single count and go, cool, we got it, we did it, we got it, let's go home. We would continue to study those systems. So being able to go back to the lagunas in Spain and characterize those sites, as well as, so Veli is working on analyzing some of the older Spanish literature where they performed microscopy analysis of these sites, like pre-sequencing, and getting an idea of how the diversity has evolved over time. Fluctuations and studying at multiple time points gives us a perspective on, okay, how much of what we're seeing is an evergreen, persistent microbial environment that will always be here, and how much is what we're seeing is something happened today. It just rained. Like something slightly different happened with the plants surrounding this ecosystem. And I think that's one of the key things.
Definitely, I agree with you, Dr. Michael. And another question, Vera, if I'm not mistaken, you actually answered this during the presentation, but if anyone missed it, you can answer it again.
Yeah, happy.
Vera says, yeah, I'm sorry. So Vera says, why is salt concentration important when studying habitability?
Yeah, so so high salt concentration and looking at environments where it's not just sodium chloride, it's other salts, like the magnesium-based salts. It's important in a Mars context because we've detected chemistries, meaning that those fluids were previously present and abundant on the surface of Mars. So by studying environments that have these high salt concentrations, it gives us potential to understand, in terms of the potential habitability of extraterrestrial sites, what would have been potential end members of those communities. But also it's intriguing to look at what's the upper limit of what microbes can survive, because we have known diversity that grows in a lab and known diversity only through sequencing studies. So it's only since we've had advances in sequencing platforms that we've been able to characterize some of these uncultivated taxa. Before that, they were just elusive and we had our limits of life where they were, and then it just takes it that one step further.
I'm sorry, thank you question and answers again. Another questions, I think again, Vera, you say what makes hypersaline environments good analogs for early Earth or Mars?
So in terms of early to slightly later on Mars, so Noachian Hesperian transition, you do have this concentration of fluid chemistries that resulted in the formation of the chloride and the sulfate-based salts. And there's detection of even more obscene salt concentrations proposed beneath the pole, the northern pole on Mars, as well as derived from chemical analysis by the Perseverance and Curiosity rovers. In terms of good analogs for early Earth, it's intriguing to see when we do things like molecular clock calculations on extremophilic taxa that many of the adaptations to high salt concentrations are present at very far back points. So like if we have the branching of bacteria and archaea, we see the persistence and presence of these traits. So it indicates that this was a key selective factor even 3.5 billion years ago.
And another question, Elif says, how can we distinguish between active life and ancient DNA in salt samples?
That's a great question. And with all things, it's so important that we have controls and caution when we go forward with analyzing different samples. I think it was in the 80s they reported potential life found in a Martian meteorite. And I think over conclusion of materials can obviously have negative effects on trust in science. Distinguishing between active life and ancient DNA, I would say if we see observed intact cells, and this is where things like entombed cells, like what happened with iron incrustations and possibly with movement of fluid inclusions within evaporitic minerals, is a problem, because there are some minerals that are very, very good at looking almost identical to entombed cells. And that's where things become more complex. And so, yeah, there has been significant claims made in the literature about where the oldest microbes have been isolated from. And that's why it's a subject of active debate, because there's always the concern that the microbe that was isolated from a salt crystal, maybe it wasn't an ancient microbe from a fluid inclusion inside, but simply a salt-loving microbe on the outside that weaseled its way in.
Oh, thank you for that great answers. And finally, questions. Hude says, how does screen electron microscope analysis support observed chemical chains?
Yep, cool. Fun question. So you're able to get these elemental maps effectively. So your sample goes under the microscope, and then you get this scanning that identifies where the different elements are concentrated. And this was how we identified that all of the sulfur in our Mars simulation experiments had ended up in the precipitate. They weren't present in the fluid anymore. It's not just enough to see that it's not in the fluid. We also had to work out where it had actually gone. So, I mean, electron microscopy is an incredibly powerful analytical technique. It also requires incredibly extensive infrastructure, which is why it would be ideal for something like Mars sample return, but obviously wouldn't be possible in the context of in situ analysis and detection.
And thank you so much, Dr. Michael. I hope we will see you again next year. We have reached the end of our time. So
thank you very much.
You are welcome, and thank you again for joining us. I hope next year again see you. So guys, we are taking a short break now. We will continue after the break, everyone, and see you later.
Hi everyone again. I'm so sorry to announce that, but we have a technical issue with the last session presentator. That's why we need to cancel that session and we need to end up this live session actually. I'm so sorry for that, but we will continue with Dr. Zachary Adam later with another webinar. So unfortunately, we need to close our video now. Before we conclude today's session, I would like to take a moment to thank my incredible team actually. As a student-led organization with no external funding, everything you have seen today, from the coordination of international speakers to the technical execution, is the result of your tireless dedication and passion. So I'm so proud of what we have achieved together. To our speakers and global audience, and thank you for being the heartbeat of this conference. Your support proves that our shared curiosity for the cosmos knows no limits. Artık Türkçe'ye geçeyim istiyorsanız. Ve tabii ki bu akşamı kapatırken benim kalbimin en büyük teşekkürü ekip arkadaşlarıma. Yavaş yavaş ekrana gelmeye başlayabilirsiniz arkadaşlar istiyorsanız. Her birinizin emeği, uykusuz geçen geceleri ve bilime olan inancı olmasaydı bu 6. yılımızda bu noktada olamazdık. Bu yüzden sizlere iyi ki varsınız diyorum. Ve değerli katılımcılarımız, online maratonumuzun ardından şimdi asıl büyük heyecan için gerisini başlatıyoruz. Yarın 26 Nisan Pazar günü saat 11 ile 17 arasında Bağlarbaşı Kültür Merkezi'nde 6. Uluslararası Astrobiyoloji Konferansı'nın yüz yüze etkinliği için bir araya geliyor olacağız. Kayıtlarımız sabah 10.30'da açılacak. Hepinizi salonun büyüleyici atmosferinde, standlarımızda ve oturumlarımızda görmek için sabırsızlanıyoruz. Lütfen geç kalmayın, yerlerinizi erkenden alın. Sizleri dört gözle bekliyor olacağız. Ve şimdi bu büyük organizasyonun arkasındaki görünmez kahramanları, sevgili ekip arkadaşlarımız zaten ekranda görüyorsunuz. Arkadaşlar söz almak isteyen varsa lütfen buyursun. Sonrasında hep birlikte el sallayarak bu akşam artık uğurlayalım.
Teşekkür ederiz geldiğiniz için, dinlediğiniz için. Seneye umarım tekrardan görüşürüz.
Evet, yarınki etkinliğimize de umarım katılım sağlayabilirsiniz. Arkadaki sesler için kusura bakmayın, yayın sırasında eğer böyle bir aksaklık yaşanıldıysa da maalesef kafenin azizliğine uğradık ama arkadaki ekibin emeğini tahmin bile edemezsiniz. Gerçekten aylar öncesinden başlanılan bir efordan bahsediyoruz. O yüzden ekip arkadaşlarıma ve katıldığınız için sizlere ve değerli hocalarıma en başta da bir kereden teşekkür ederek yayınımızı bugünlük noktalamak zorunda kalıyoruz diyelim.
Arkadaşlar ben de çok teşekkür ederim. Yaklaşık 6 senedir bu astrobiyoloji konferansını gerçekleştiriyoruz. Ben bu konferansları yaparken 20 yaşındaydım. Şimdi 25 yaşıma geleceğim artık. Umarım önümüzdeki senelerde daha dolu dolu, daha farklı etkinliklerle tekrar beraber oluruz. Bugün gerçekten bilimin dışında farklı coğrafyalardan bir araya gelerek çok güzel bir yolculuğu birlikte deneyimledik. Bu yolculuğun bir parçası olduğunuz için her birinize yeniden çok teşekkür ederiz. Hepinizi yarınki Bağlarbaşı Kültür Merkezi'ndeki etkinliğimize bekliyoruz. Bu şekilde.
Ben de bütün katılımcılarımıza teşekkür etmek istiyorum. Bu çalışkan ekiple ben de 4 yıldır çalışıyorum ve birlikte gerçekten harika işlere imza attık ve bundan sonrasında da çok daha iyi işlere imza atacağımıza içtenlikle inanıyorum. Bizim için çok çabaladığımız, emek verdiğimiz bir konferansı yarın da artık yüz yüze yaşamaya geçeceğiz. Herkese tekrardan teşekkür ediyorum. Umarım herkesin çok keyifli geçmiştir ve yarın da öyle geçecektir.
WhatsApp'ta kalmaya devam edin. Eğer yarın yüz yüze katılacak olan arkadaşlarımız varsa bütün duyuruları, gelişmeleri veya şeyleri oradan size iletiyor olacağız arkadaşlar.
Yasemin, senin söylemek istediğin bir şey var mı? Sesin gelmiyor sanırım, emin değilim ama. Arkadaşlar ilk yüz yüze etkinliğimiz olacak yarın. Bizi bugün de yalnız bırakmazsanız çok sevinirim. Berra'nın dediği gibi biz bu yolculuğa başladığımızda pandemiydi. Online'lara öyle başlamıştık. Ben o zaman lisans öğrencisiydim. Şu an doktora yapıyorum. Yani düşünün hani 6 yıla neler sığıyor. İnşallah sizler de o günleri görürsünüz. Belki ekibimizin de bir parçası olursunuz diyorum. Hepinizle o zaman görüşmek üzere diyorum. Yarın Bağlarbaşı Kültür Merkezi'nde. Şimdilik hoşça kalın. Biz el sallayarak veda edelim o zaman. Arkadaşlar you.